What's up, Ninja Nerds? In this video today, we're going to be talking about anti-neoplastic agents, and this is going to be a monster of a lecture. The reason why is it's going to combine aspects of step 1, but it's also going to really talk about more of the step 2, more of the decision making, more of the therapeutics, more of the adverse effects. But I think in order for us to understand these things, we have to build up a little bit of foundation in the physiology. So we'll talk about the cell cycle briefly, we'll talk about the cell regulation, and then we'll talk about some other kind of immune and angiogenic kind of influences as well. Then what we'll do is we'll go into each individual kind of anti-neoplastic agent, talk about their names, talk about how they work, and then what we'll do is we'll go into the indications. Why do I give this one? Why do I give this combination? And then lastly, we'll talk about what are some things to monitor for because of the potential for the adverse effects and toxicities of some of these kind of anti-neoplastic agents. However, this is going to be a really long lecture. If you want like a quick little summary or a little recap of all of this kind of the more step 1 content, you can go down in the chapters and you'll be able to go to the little summary part and you can kind of watch the whole summary, which is going to explain most of the video. And then we're going to start talking a lot about the indications and the adverse effects. All right, let's get started. All right, my friends, we're not going to go crazy. We talked about this more in our cell cycle video. If you guys want to watch that video, go and click on that video. It's going to be in our cell biology playlist and we go over the cell cycle in a lot more detail. This is going to be more of just kind of a quick little reminder, if you will. So when we talk about the cell cycle, let's say that we have a cell, right? You just basically took a cell, you divided it into 2 daughter cells, right? And that's going to be through a specific phase called the mitosis or the M phase. But once you have this cell can either kind of, it has 2 options. One is it can go into a resting state, right? And so we have a phase here and we call this phase the G0 phase. And this is basically the phase where, I'd say in the most simplistic way possible, this is the quiescent phase or the resting phase. This is where most of your cells are basically at rest. They're not kind of actively replicating and dividing. They're mature, differentiated cells. That's going to be one of those phases. An interphase, really, we'll talk about the different components, but one of those we kind of consider the G0 phase or that quiescent area where the cells are resting. They're not really constantly actively replicating. They're more in their mature, differentiated states. Another option is this cell, right, what it can do is it can start kind of taking and getting ready to undergo DNA replication, right? And so let's say that, for example, here I have, a male chromosome, and then we have the female chromosome, right? And then you also have a bunch of other things, proteins and enzymes and organelles. When I want to take this cell and get it ready to divide, I need to start getting a lot of enzymes that are needed for kind of the whole DNA replication process. I also need to start getting organelles to be built up so I can share the organelles between 2 cells. And so that's what usually happens going from this part to this part. And so that's what we would see is we would see kind of the same number of chromosomes. We haven't replicated at this point, but we may have more proteins. We may have more enzymes that are needed for the DNA replication process. We may have more organelles. This phase here is called the G1 phase. So it's called the G1 phase. All right, so that's the next part of interphase. So we have the G0 phase, which is the quiescent one. It's the resting state. That's where most of the cells are in the rest. They're not actively kind of replicating. They're mature, differentiated. G1, they're getting ready to start kind of preparing for the replication process. They're making more organelles. They're increasing proteins and enzymes needed for DNA replication. Then what's going to happen is we're going to take this cell and look what changes here. We go from having this part here and we duplicate our chromosomes. When you go from this cell to this cell and you duplicate your number of chromosomes, you underwent the DNA replication process. And at this stage, this is called the S phase. So this is where DNA replication takes place, right? A lot of this should sound familiar from your basic biology, right? Then what we're going to do is we're going to take this cell, which has these kinds of extra, now in this case, we've kind of duplicated the DNA. We need to start getting these kind of cells ready to share the DNA. So I'm going to start increasing the cell size. I'm going to start getting the cytoplasm bigger. I'm going to start making more of the enzymes that I need to help me to start separating this cell. And that's when we get ready to go into this next stage. And this is called the G2 phase. This is called the G2 phase, right? The G2 phase. And all that's happening here is you're kind of just getting things ready. You're kind of getting the cell ready. You're increasing the number of proteins and enzymes that you need in order to separate this actual kind of chromosomes into the separate daughter cells. but you're getting the cytoplasm bigger, you're increasing your enzymes, and you are getting ready to start dividing these cells. Then what happens is we're going to take the cell and we're going to start dividing the actual genetic material. And when we divide the genetic material into two identical daughter cells, that is when we have what's called the M phase. All right, so this is the M phase. Now the M phase is also referred to as mitosis. Mitosis consists of a couple different parts. So you can oftentimes remember this via what's called PMAT, right? And so we call this prophase, right? Then we have what's called metaphase. Then we have anaphase. And lastly, there is telophase. And again, we talk about this more in the cell cycle video, but for simplicity's sake, all that we do in the prophase is we start kind of condensing down this genetic material. We condense it down. And then also, you start to begin to see these things called, and I'm going to represent them with circles and maybe some lines coming off of them. Your centrosomes, which are going to be where the mitotic spindles originate from, they start kind of going to the opposite poles here. Then, in metaphase, what happens is this chromatin is pretty condensed, and then you start noticing that these mitotic spindles are moving, and then your chromosomes are lined up in this nice kind of like equatorial plate, and we call that metaphase. Then, you have the point here where the mitotic spindles are starting to depolymerize, and they're separating these sister chromatids into different cells. And then we get into telophase where we're literally starting to cleave the cells in half. This is the different parts. Now the reason why I'm stressing on all of this is when we give antineoplastic agents, they can act in different parts of the cell cycle. So we can have drugs inhibit the cell when it's in the S phase. We can have drugs inhibit the cell when it's in the G2 phase. We can have it inhibit when it's in metaphase and anaphase. And that's the important point. So we'll talk about this a lot, but a lot of the drugs, they tend to really have the most profound effect in the S phase, when the DNA is replicating. We have some of them that work in the G2 phase, as the DNA is replicated, but we're getting the actual cell ready to separate. And then metaphase and anaphase tend to be some heavily involved areas. So this is what I want you guys to understand, because now what we'll do is we now build up a point here where I can say which drugs are antineoplastic chemotherapeutic agents that are cell cycle specific, meaning it involves one of these phases, and which ones are cell cycle non-specific, meaning that they can affect any point, including the G0. Because oftentimes when we talk about, and this is one of the key basics of chemotherapy, when you talk about cancer cells, they have a high growth fraction. They replicate like bunnies. When you give chemotherapeutic agents, there's something called the log-kill hypothesis. Write this down. The log-kill hypothesis essentially says when you give a chemotherapeutic agent, it causes a constant fraction of cells to die off with every round of chemotherapy. It's not a constant number of cells. It's a constant fraction of cells. And that type of cell that is usually getting killed by chemotherapeutic agents are highly replicating cells. That's what chemo goes after. What are the most highly replicating cells in our body? Think about it. If you have cancer, it's cancer cells. What's the one that if we didn't have this thing proliferate, you'd see the person's bones and muscles and fat? Your epidermis, so your epithelial tissue, that makes your skin and all of your mucosal linings. So you ever notice how when people take chemotherapeutic agents, what happens to their parts of their skin? What actually makes up part of what's actually technically skin? Hair. They have hair loss, right? Another thing, it affects their mucosa, so they can develop mucositis. They can have a lot of GI problems. You ever notice some common symptoms of chemotherapeutic agents? Nausea, vomiting, diarrhea, abdominal pain. The other thing that it also affects is your bone marrow. Your bone marrow is constantly replicating and making different types of cells. If I give chemotherapeutic agents, what would it affect? It would affect those. And so sometimes they can have decreased numbers of white blood cells, red blood cells, platelets, so pancytopenia. And so that's why it's really important when we talk about these drugs, you should have cell cycle specific. They're affecting cells at different parts of the cell cycle, or cell cycle non-specific. They can affect it at any stage, and that includes the ones who are in the resting state. That's your cell cycle non-specific. Okay, now, we've kind of built up an idea that we know the different phases that the cell can be in. And so whenever you have a cancer cell, these cancer cells are scary, man, because they can basically develop mutations that allow for them to keep going through the cell cycle and replicating and replicating and replicating. All right, so they develop mutations for whatever reason. Those mutations allow for them to increase the proliferative genes and shut down the tumor suppressor genes. The other thing that comes into this is now talking a little bit about how we regulate the cell cycle. This is really critical. So let's say, for example, here we have the DNA inside of a cell. There's different ways that when we kind of go through these phases, and this is a critical checkpoint. Usually when you go from the G1 phase into the S phase, there's a checkpoint there. And there's a very special set of enzymes that kind of control this step. And these enzymes are really important, so let's write these down. This one is actually, it's kind of a group. So you have cyclin D, and then you have these CDKs, which are cyclin-dependent kinases. So you can't have the CDKs without having the cyclin D. But usually this is going to be CDK 4 and 6. All right, I do want you to remember that. The other thing that you'll have is this red protein. This is called retinoblastoma protein. We're going to write that as RB. And then the green one is called E2F. Now, what normally happens is that this is influenced by a bunch of different upstream pathways. And so what CDK4 and 6 is supposed to do is it's supposed to put some phosphate groups onto retinoblastoma. So it's supposed to, basically, it's supposed to kind of add phosphates. Let's say that I add some phosphates here. When you add phosphates to the retinoblastoma protein, guess what it does? It lets go of E2F. So it basically, once the phosphates bind to it, boom, E2F gets released. So now we're going to kind of dispense the E2F. So here E2F was bound. Now I need to come over here for a second, guys. Grab my green marker. This is going to be my free E2F. So now I have free E2F. And this is actually going to start moving over. And then what it's going to do is it's going to act on kind of special different enzymes. And let's say that it acts on some special enzymes, and when it, special genes, and it basically is going to stimulate genes. And let's say that these genes want to kind of push the cell into the proliferative state. It wants to push the cell to go from here to here. That's what it can do, right? So we're going to activate different types of genes to really help to stimulate this process. That's how this works. So CDK4 and 6 phosphorylate retinoblastoma, release E2F. E2F activates particular genes because E2F is a transcription factor. When it activates these genes, it then says, go ahead, cell, continue into the actual cell cycle. That's pretty cool, right? And so this is a very key regulatory point. The key thing behind this is that there's a lot of upstream things that are influencing this. All right? So if a cell wants to go through that cell cycle, it needs these different points where we can say, okay, you're good. Here's a couple different things. One is we can have something called well, there are different types, but we have what's called usually it's a tyrosine kinase receptor. So you have different types of tyrosine kinase receptors, right? So many different types, right? And so usually what happens with these tyrosine kinase receptors is you have some type of molecule, a ligand of sort, that will bind onto these. Usually these are some sort of growth factor. That's usually the key. There's some sort of growth factor. And when you have this growth factor, what it'll do is it'll bind onto these receptors. And there's a couple different types. I mean, we'll list them. One of them is called EGFR, the other one's called ALK. So you have one called EGFR. You have another one called ALK, right? And there's even, not even membrane-bound ones. You have another one too, which is actually inside the cell. It's kind of like a tyrosine kinase, but it's a non-membrane-bound one. So it's really an interesting one. And this is called a BCR-ABL. So this is called BCR-ABL tyrosine kinase. All right? And these are all like special enzymatic like receptors. This one is membrane bound. This one is kind of like in the cytoplasm. Either way, when you have something like this happen, like let's say that you have a growth factor hit the tyrosine kinase receptors, it can trigger intracellular pathways. Usually there's a couple different pathways, and I don't want to go too crazy, but they can increase the presence of some different kind of intracellular processes called like PI3K, AKT. It can do another one, which is actually really important. It's called the RAS pathway. And so we actually call this the MAP kinase pathway, which is basically you increase the activation of RAS, RAS then leads to the activation of RAF, RAF leads to the activation of MEK, and MEK is usually going to lead to the MAP kinase. Either way, these are intracellular processes. And the other thing is it can increase the presence of what's called STAT, which is a signal transducer of activated transcription. Either way, growth factors bind to these receptors. They increase this pathway. This pathway then does what? It then goes and increases the activity of specific processes. Like what? Well, it can lead to some downstream processes. So here's CDK4 and 6, right? It's heavily influenced. It may lead to these downstream processes that lead to a stimulation of CDK4 and 6. And so if I stimulate CDK4 and 6, what will it do? Phosphorylated retinoblastoma. What would that do to E2F? Let go of E2F. If I let go of E2F, that'll activate specific genes. These genes will then push that cell to go through the cell cycle and replicate. So you see how growth factors can do that? We can come up with drugs to target these, to target the EGFR receptor, to target the ALK component of the tyrosine kinase. So we can do that. The other thing is the BCR-ABL. You can have specific things, particularly ATP-bounded sites. And what you can do is you can block this little component here, because BCR-ABL, if it gets activated, when it's stimulated, it can also trigger these pathways, the same kind of thing. And so if I have a drug that could bind into the ATP binding site of this, I could inhibit it, and I could shut down these pathways and shut down this going from G1 to the S phase. That's cool, right? The other thing we have, not only do we have these tyrosine kinase receptors, but we have other receptors too. So for example, we also have ones called Janus kinase. So these are usually called your JAK2 receptors. And what happens is these usually combine some type of growth factor, same concept. Let's actually draw these kind of like coming over here as well. So you have growth factors binding here onto this, and you have growth factors coming over here and binding onto this. Guess what? They can do similar things. The primary one that they tend to lead to the activation of is STAT. Either way, same downstream process. So if I had a drug that could block this, I could potentially shut down this kind of cell proliferation. That's why we usually give this kind of drug in people who have myeloproliferative disorders. We give a lot of these drugs, particularly the EGFR and the ALK ones, we give these things in like lung cancer. We can also use these a lot in certain types of hematologic malignancies, especially things like for this one, CML. But we'll talk about that. Another one is you can have something called a HER2. All right, so a HER2 is like a human epidermal receptor. And this one can also be bound by different types of ligands. And when that leads to this process, again, it leads to the same thing. It can, and we'll just kind of write this down here. We'll do the same thing. But if you activate this receptor, guess what you're doing? You're increasing the PI3K, AKT, increasing the MAP kinase, and increasing the STAT signaling. So PI3K, AKT, you're increasing the MAP kinase pathway, and you're increasing the STAT pathway. Either way, all of these things are leading to that downstream influence on CDK4 and 6. So if I gave a drug that could block this receptor, I may shut down this pathway. And that's a concept there. Another thing that I also want you guys to understand is we can have receptors that are present inside of the cell. Inside. So for example, you can have different types of receptors. This could be an estrogen receptor, right? This could be, you have other processes too. It could be an androgen receptor. And if these things come inside and bind to these receptors, guess what? They can also lead to this process here. And so what I'll do is I'll kind of bring this one here and I'll join in. I'll make this a little bit smaller. We'll squeeze this one in here. But it's the same concept. If you have estrogen or androgens binding onto these receptors, once you do that, this is going to lead to them activating the CDK4 and 6. So this is why I'm trying to tell you this. If a patient has scenarios where they have abnormalities or mutations in the tyrosine kinase receptors, abnormalities in their JAK2 receptors, abnormalities in the HER2, abnormalities in the estrogen and androgen receptors, or maybe in pathways that influence that, because there's other ways. You have other enzymes like aromatases, which are also supposed to convert estrogen into androgen. There's so many different types of targets. The big thing is that you have all these upstream pathways that are leading down to this pathway. And this really helps to drive the cell cycle. If I could come up with ways of targeting all of these different steps, I could shut down a lot of the cell proliferation. That's what I need you guys to understand, is if all of these are really coming downstream and influencing this, if I come up with drugs to target this, I provide a massive benefit. And guess what? I can even have drugs that influence these pathways. So that's what I'm trying to get you guys introduced to, is understanding the basic pathway and how we can bring drugs in to target this. And eventually all they're gonna do is shut down CDK4/6, shut down phosphorylation of retinoblastoma, shut down E2F, and shut down the initiation of that cell cycle. So if we think about this again, kind of giving you a little recap, there's a couple different types of receptors I want you guys to know. I think a really, really important one is the tyrosine kinase. Do not forget these. So these are your tyrosine kinase, and these could be the membrane bound, but they could also be the constituent enzymes like the BCR-ABL inside of the cell. Please don't forget that. The other one is going to be the JAK2. The other one is called the HER2. And then the last one is going to be your steroid receptors. These are your steroid receptors. Usually this is for things like estrogen, androgens, etc. All right? Pathways that are usually downstream from this is going to be things like PI3K, AKT, and some of you who are actually really into your kind of like cell biology, you know that this will lead to the increased production of mTOR, which we have certain chemotherapeutic agents as well. You have, we'll write this one out, the RAS, RAF, MEK. This is usually via the MAP kinase pathway. And then lastly, you have the STAT signaling, right? Basically, all of these in some way, shape, or form increase these pathways. I can shut down this, and I can shut down all of these. The ones that usually are the most talked about for the step 2 is going to be this one. And then lastly, we have a lot of enzymes that are controlling in this process, but the key ones that I wanted you guys to really know that influences this is what? CDK4 and CDK6. There's a reason I'm writing all of these down. And the reason why is because when we start talking about each one of these drugs, we're gonna have drugs that influence the S phase. We're gonna have drugs that influence metaphase, anaphase, G2 phase. We're gonna talk about those. Cell cycle specific, cell cycle non-specific. When we get into the drugs that influence cell cycle regulation, we're gonna have tyrosine kinase inhibitors, JAK inhibitors, monoclonal antibodies. We're gonna have different hormonal therapies. We're not gonna talk about these, but we're gonna talk about some specific types of drugs that work again in this pathway, the RAS, RAF, MEK, MAP kinase pathway. And then again, we're gonna have drugs that are targeting the CDK4 and 6. All right. Now that we've done this, there's one more thing that I gotta talk about before we really start getting into these drugs, and that's, guess what? These cancer cells, not only do they gain the capacity to have mutations and replicate uncontrollingly, or they have things that are driving them to really go and undergo a lot of cell replication, but we have to have ways of controlling these cancer cells, and we have immune surveillance that we're gonna talk about next. All right, so now we're gonna move into the next part. So this is gonna be the immune evasion. So it may seem like this is very random. I promise there's a method behind the madness, if you will. So the reason why I'm kind of doing this little kind of extra piece is because there's a lot of newer drugs that we're using as antineoplastic agents that can really essentially activate our immune system, turn the T cells on to start kind of fighting some of these cancers for us. One of the interesting things here is that whenever cancer cells, not only do they gain this ability to, develop mutations, and then because of those mutations, they allow for alterations in their cell cycle. And so because of that, they may increase proto-oncogenes, which allows for them to proliferate, shut down tumor suppressor genes, which again evades apoptosis, so they can replicate, and then they don't die. On top of that, they're also really, really kind of smart in the sense that they can potentially evade the immune system. And so they can do that by increasing the expression of specific ligands on their surface that shuts down T cells, or they can increase the expression of certain ligands on the T cell, which then makes it where it can't interact with antigen-presenting cells to activate them. So they're kind of interesting in that sense. And then the other thing is that cancer cells also, they want to steal all of the blood supply so they can get oxygen, nutrients, they can grow, they can metastasize. And so they have a lot of angiogenic mechanisms that are involved. So when we talk about cancer cells and T cells, let's kind of take a look here. There's different receptors on their surface that need to kind of interact essentially. And so one on the cancer cell, usually they express something called a major histocompatibility complex, usually with some sort of like antigen on the surface. In order for that to really take place, you need to have the T cell receptor interact with the antigen, and then usually some type of cluster differentiation protein of some sort. We'll just kind of represent this with like, let's just use for right now, we'll use this baby blue color. So they usually have some type of cluster differentiation protein which interacts with the MHC, right? Now, the other thing here is that on the T cell expresses something called PD-1. So it expresses something called PD-1. Now, usually PD-1 has to interact, it usually will interact with something called PD-L1, which is the ligand, and that's usually something that cancer cells are smart. They will increase their expression of PD-L1. If they increase their expression of PD-L1, that allows for this interaction to take place. So then what you're going to do is you're going to allow for this interaction to occur. If you stimulate this interaction, guess what it does to the T cell? It tells the T cell, hey, go ahead and turn off, buddy. You don't need to do anything to me. And it kind of shuts the T cell's activity from killing this cancer cell down. And so that's kind of interesting, as it can basically, in a way, it can turn that T cell off. So let's say here, we'll just kind of write it like this. It turns off the T cell. All right, the next concept is that cancer cells also are interesting because what they can do is they can, in some way, signal to T cells. And they can cause these T cells to increase their expression of another type of molecule. So again, let's say here we have a T cell. Here you have an antigen-presenting cell, right? This could be a macrophage, dendritic cell. It could be a bunch of them. On their surface of the T cell, you have what's called a what? A T cell receptor. Here on the antigen-presenting cells, they express MHCs. Usually this is a 1, this is a 2. There's an antigen. And then again, technically for this true interaction to take place, you need a cluster differentiation. Now, in order for this T cell to be activated, it needs not just this interaction between MHC and CD, TCR and antigen, it needs co-stimulation. That co-stimulation is done via something called B7 on the antigen-presenting cell and CD28 on the T cell. Now, what's interesting is that these cancer cells will tell the T cell to increase the expression of another molecule. And this molecule is called CTLA4. Now, why is this a problem? When you have CTLA4 and you're increasing the expression of it, it beats out the interaction between CD28 and B7. That's what ends up happening. So then what ends up happening here is if I kind of like move this, let's say that this guy becomes so more like a strong affinity, it essentially will move this CD28 out of the way. And then this CTLA4, guess what it's going to do? It's going to come over here and it's going to bind in. That's one of the benefits here. So here's CD28, and here, my friends, is CTLA4. And now these can interact. When this right here occurs, this interaction occurs, it basically turns the T cell off. So now, if I do this, I turn off the T cell. And if I turn off the T cell, can it go after the cancer cells? No. Now you're probably like, okay, what's the point of all of this? I get it, we're gonna kind of come up with drugs, right? Yeah, if I can have a way of inhibiting CTLA4 or inhibiting these two, maybe PD-1 or PD-L1, I could potentially do what? Turn the T cell on to kill the cancer cells. Turn the T cell on to kill the cancer cells. That's what we'll talk about eventually when we get into those drugs. And we'll talk about these, they're referred to as your immune checkpoint inhibitors, which are like types of monoclonal antibodies. All right, the other thing that's really important about cancer is, again, the angiogenic effect. Blood flow is key. You know, cancer cells, they need oxygen, they need nutrients, and they require a lot more because of how heavy their proliferation process is. They need a lot of these metabolic processes to be driven by oxygen, and that needs to be present to make ATP. And so because of that, cancer cells are smart. And what they do is they secrete a chemical called VEGF, right? So VEGF is vascular endothelial growth factor, right? So that's what it's called. It's called vascular endothelial growth factor. Now, vascular endothelial growth factor will basically bind onto these receptors, and these are called your VEGF receptors. What will happen is when VEGF binds onto these receptors, where is it present? That's the first question we should ask. Do you notice where this is? This is the blood vessel, but what part of the blood vessel? The endothelial cells. And so what it does is it activates the endothelial cells to undergo replication. And when they undergo replication, it goes from this vessel to this vessel. Do you see all these sprouts that came off? Because the endothelial cells, they started sprouting off. And they make more vessels. What's this called? Angiogenesis. So what it does is it increases, let's actually write this here, it increases angiogenesis, which is what? Making more blood vessels. Now, if I make more blood vessels, what's the benefit of that, my friends? I'm going to increase oxygen delivery and nutrient delivery to the cancer cells. So therefore, you get increased O2 delivery. If I get increased oxygen delivery, nutrients delivery, what's going to happen to these cancer cells? They're going to grow. And if these cancer cells grow, my friend, oh boy, that's when we get some problems. So if we had a way to be able to come up with drugs to target these, especially VEGF, I may be able to shut down the angiogenic process and shut down the oxygen delivery and shut down the cell growth. That's kind of really interesting. So at this point, we've talked a lot about the basic physiology. What I now want to do is let's take it one step forward and say, let's integrate the drugs into these basic physiological processes and see how we can influence these processes, see how we can shut it down. And I think it'll help us to remember these things a little bit easier. All right, so let's move on now to the cell cycle inhibitors. So we talked about the cell cycle in great detail, right? And again, it's really, it's pretty simple, right? When you take and you actually take a cell who you've duplicated the number of chromosomes, you want to separate those out to your daughter cells, right? And so these should have the same number of chromosomes. And again, that was denoted as the M phase. That was your mitosis. That was the prophase, the metaphase, the anaphase, the telophase, right? Then if we take one of these cells that just got duplicated, it's going to want to get duplicated again. So what does it have to go through? Well, it can either go into a resting state or it can start growing and getting ready, making enzymes, proteins that we need to replicate the DNA. So that's what we do. We make more organelles, we make more proteins, and then we get ready to duplicate the DNA. That's when we go into the S phase. Once we've duplicated the DNA, we want to start making more proteins, more enzymes, increasing the cytoplasm size so that we can split this equally between the two cells. That's when we go into the G2 phase. We talked about this, right, with the cell cycle. There's drugs that are targeting specific points of the cell cycle. When we have cancer cells, we want ones that are constantly going through the cycle. We don't want a lot of the cells to be in this resting state. All right, that's the important thing. And so we have cell cycle specific. So what does that mean? Meaning that they target G1, S, G2, M phase. That's where they're working, G1. They're working particularly in the S phase. They're working in the G2 phase. They're working somewhere within the M phase, right? That's where they're working. Whereas cell cycle non-specific, they can occur anywhere. Usually when we say cell cycle specific, we have S phase inhibitors, G2 phase inhibitors, M phase inhibitors. When we talk about cell cycle non-specific, this could occur, dude, you could even do it in the resting state. It could be G1 and G2. I could have it occurring in the S phase and the M phase. So this is not specific. It's not a specific area in the cell cycle that they're inhibiting. That's the difference between these two. And this is the key one, is that these cell cycle non-specific can occur in that resting state, that G0 phase. Whereas this, it's not going to really affect the G0. And usually it's going to be one specific phase. So for example, let's kind of make sense of this. We have the S phase. This is a really important one. So the S phase inhibitors. All right, so we'll talk about it in this sense, but we'll get into more detail on how exactly they inhibit the S phase. But when we talk about S phase inhibitors, we have a couple different drug categories. The big group, and we'll talk about a couple of them, is going to be your antimetabolites. These are a heavy class of drugs. And when we talk about these, we're just kind of introducing it a little bit. We're not going to go in crazy detail. How they really work is they inhibit nucleotide synthesis. So they may inhibit the formation of purines, pyrimidine nucleotides. They may inhibit their incorporation into DNA. They may inhibit some of the enzymes that are needed to make the DNA from those nucleotides. That's how they are working. Another group of drugs that also work within the S phase, and again, S phase is when the DNA is replicating. You're making more DNA. Topoisomerases come into play. You guys remember that from cell biology, right? We have topoisomerases. Basically, you have the DNA replication fork downstream. Upstream from that, you have an enzyme because you have what's called torsional stress. And so whenever you have these torsional stress, what happens is you have supercoils, and then topoisomerases are supposed to come and actually cut the DNA, let it unwind, reduce that torsional stress, and then connect it back together. What if I had a drug that could cut and then let it unwind and never glue it back together? The thing would unwind, and eventually you wouldn't be able to allow for the DNA to completely heal, and the replication forks would be all jacked up. That's going to be topoisomerase inhibitors. And there's 1. And 2, this is the one that only works in S phase. So topoisomerase 1 works in the S phase. Therefore, topoisomerase 2 works in a different one, and we're going to talk about that. Usually it's the G2 phase. So let's move on to the next one. The G2 phase inhibitors, they only work particularly in this phase. Well, again, I kind of already hinted at one of them, and that's going to be your topoisomerase 2 inhibitors. There's another drug, and this is a part of your anti-tumor antibiotics, which we'll talk about. So topo 2 inhibitors. And then the other one is going to be bleomycin. Now this is an anti-tumor antibiotic. We'll talk about that a little bit later, all right? The next one is M phase. So M phase inhibitors, which is going to be working in mitosis. So M phase inhibitors, they're primarily affecting things like your metaphase and anaphase. And so what they're doing is they're inhibiting particularly the microtubules from polymerizing and depolymerizing. And this is going to be your microtubule inhibitors. And we'll talk about these groups a little bit later as well. So microtubule inhibitors. Now the last one I'm hesitant to kind of include because technically it's not really a phase, it's more of a checkpoint. So usually you have something called checkpoints. And there's a really important checkpoint which occurs right here, kind of before we go into the S phase. This is called the G1 S phase checkpoint. And so when we talk about this G1 S phase checkpoint inhibitors, there's different cyclin-dependent kinases that regulate the cell cycle. That's why I want to talk about it more over there. But we had a specific enzyme that's really involved in this process. It's not upstream, no kind of upstream involvement. I'm talking about only direct. And that's the cyclin-dependent kinases. So technically we could say that a CDK4 and 6 inhibitor could potentially affect the cell cycle, specifically around that checkpoint level. But I'm going to talk about that a little bit more over in the cell regulation processes. All right, but for right now, we're just going to kind of slowly integrate it here, but be aware we will talk about this more so when we get into the cell regulation. All right, cool. So here's the drugs that specifically inhibit parts of the cell cycle. These could occur at any phase. It's not specific, dude. It's just not. So because of that, we're going to talk about these. This is usually going to be, I'd say, 2 primary categories. One is called your anti-tumor antibiotics. So your anti-tumor antibiotics. You're like, wait, what the heck? Anti-tumor antibiotics? These are weird. These are going to be some ones that we use a lot called doxorubicin, daunorubicin. We also use dactinomycin, mitomycin, and bleomycin, funny enough, as an anti-tumor antibiotic. But it's the only one that is specific. All these other ones are usually non-specific. The other one are called your alkylating agents. And these have different kinds of groups that we'll talk about as well. This is going to be things like your nitrogen mustards like cyclophosphamide, ifosfamide, melphalan. We'll talk about your other ones, which are your platinum compounds, cisplatin, carboplatin as well. But these are going to be very, very particular in that they do not have a very specific cell cycle that they influence. Now, we'll talk about this as well a little bit later, but when we think about the process of how when we take something and we form something like proteins, we go through this very consistent sequence where we start off with usually things like nitrogenous bases, right? So this could be things like your purines, or like pyrimidines, right? In this case, we're using something like purines. Then the purines are making something called ribonucleic nucleotides. So basically we're taking the nitrogenous base, adding on a carbohydrate group and a phosphate group. And then we convert that and we actually kind of dehydrate it. So then we make something called a deoxyribonucleotide. And then we take these deoxyribonucleotides, right? Which is, usually it's a form of, we take these things like adenosine, thymine, cytosine, all of those guys, and we're incorporating them into the structure and then adding it into the DNA. And so this is how we generate things like DNA. And then DNA we can translate, and we can turn this into RNA. And then RNA, we can do, we can transcribe DNA, make RNA, and we can translate RNA to make proteins. So we have drugs that can work along this process. So you can think about them as being cell cycle specific or non-specific, but you can also think about this mechanism as well, because we're gonna have a bunch of different drugs that are influencing all of these different stages. Let's start off with talking about the antimetabolites first. So when we talk about antimetabolites, they're really going to be affecting, I say, a couple of those stages up there. They're going to be affecting going from like the nitrogenous bases to the nucleotides, the ribo, going from ribo to deoxy, going from deoxy to DNA. So we have a couple different groups. One is called folate antagonists. Now, folate antagonists really consist of one drug, and this is called methotrexate. All right, this is called methotrexate. Now, a little intro into this process that occurs inside of our cells. Let's say here's a cell. We need this cell to start incorporating nucleotides into the DNA or to make DNA. So we want to make some DNA for this cell so that we can actually help to make another cell. Well, we need a molecule called folate. Folate's pretty crucial. And so what happens is folate gets brought into the cell, and then what happens is eventually it gets converted into something called dihydrofolate. And then dihydrofolate can be converted into another molecule called tetrahydrofolate, right? So we're going to convert dihydrofolate into tetrahydrofolate. Then tetrahydrofolate gets converted into another molecule called 5,10-methylene. 5,10-methylene. Tetrahydrofolate. Now here's the important thing here. When 5,10-methylene tetrahydrofolate is formed, it can go a couple different processes. But the important one is that it can actually get converted back into dihydrofolate. When that happens, this interaction creates more of a donation effect to help us make something else. And that something else that we're going to make is we're going to take a deoxyribonucleotide called DUMP, which is basically that kind of deoxy form of uracil, and convert it into DTMP, which is basically the deoxy form of that thymine nucleotide. Guess what this is going to do? This is going to help me to make thymidine. Thymidine is a very precious nucleotide that we need to be incorporated to make DNA. So this is going to help us to make the DNA. Now, I have basically a couple different reactions here. Let's use one enzyme here. This is regulated, this step here is regulated by an enzyme called thymidylate synthase. All right, I'm going to abbreviate this TS, thymidylate synthase. There's another enzyme, let's do this one. Well, we'll do blue color again. This one is called dihydrofolate reductase. What is this enzyme called? This is called dihydrofolate reductase enzyme. Now, the drug methotrexate, which is kind of cool here, we're going to abbreviate this MTX. MTX is going to work by, it gets taken into the cell and inhibits this enzyme. If you inhibit the dihydrofolate reductase, can you convert this reaction? No. If I can't convert this reaction, what happens to my tetrahydrofolate levels? They drop. What happens to my 5,10-methylene tetrahydrofolate levels? They drop. Can I now, even if I have this in excess and I have this enzyme, can I now convert this into this DTMP? No. So what happens to this process? It gets shut down. Now I have less of the thymidine. If I have less of the thymidine, can I make the DNA? There's a decrease in DNA synthesis. So now I shut down that DNA synthesis. Oh man, that's cool, right? Another thing here is tetrahydrofolate is also used to make other nucleotides. We're not going to talk about this pathway. We talk about it in the nucleotide metabolism video in our biochemistry playlist. Go watch it there. But this can also be used to make things like purines. And so if we have less of this tetrahydrofolate, we theoretically shut down the synthesis of purines as well. And purines, this is a pyrimidine, right? So thymine, usually the thymidine is a pyrimidine. Purines, they're also going to be involved to make DNA. And so that'll shut down your DNA synthesis as well. But the primary one I want you to remember is thymidine. So when we give drugs like methotrexate, it's inhibiting this enzyme and shutting down this process. Now, another thing that's really, really important here is the pyrimidine antagonist. So now we gotta talk about the nucleotides. So we kind of introduced it a little bit. Pyrimidines are going to be the single ring. So when you think about that, that's kind of, I just remember it's the 6-ring structure, right? Purines are the double ring structures, right? So pyrimidines, this is usually going to be things like your thymine, your uracil, and then your cytosine, right? Whereas your purines are going to be your adenosine, your guanine, it's going to be those guys. Now, I want to have specific drugs that can inhibit the formation of pyrimidines and their incorporation into the DNA. So there's a couple different drugs. One of those drugs is called 5-FU. No, FU. No, I'm just kidding. So 5-fluorouracil. Another one is called cytarabine. So it's called cytarabine. And then one more that's kind of an important one is called gemcitabine. So gemcitabine. So these are all different types of drugs that we can utilize as pyrimidine antagonists. And all I want you to think about is, for the most part, some of these act like a nucleotide, but they're not a functional nucleotide, which is kind of interesting. All right, let's go over 5-FU first. So 5-FU is it kind of goes off this pathway already, so this is kind of nice. We take something like folate, and folate gets brought into the cell. When it gets brought into the cell, what does it get converted into? Dihydrofolate. Dihydrofolate gets converted into tetrahydrofolate. Tetrahydrofolate into 5,10-methylene tetrahydrofolate. 5,10-methylene back to dihydrofolate. And then we use a molecule called what? DUMP. So we use something called DUMP and convert this into DTMP. And this is utilized to make something like DNA, right? So now, with that being the case, we have a couple different enzymes involved here. So this one here is called dihydrofolate reductase. We talked about this one. What's this one here? The thymidylate synthase. This is called the thymidylate synthase. Guess what 5-FU does? The 5-FU shuts down thymidylate synthase. So that's what 5-FU is going to do. It's going to get inside of the cell, right? And when it gets in here, oh, I got to bring this guy somewhere around here somehow. We'll bring it here. There you go. It's going to inhibit thymidylate synthase. And so if it inhibits thymidylate synthase, you won't allow for this interaction to occur. And if you can't allow for this interaction to occur, DTMP levels drop. If DTMP levels drop, then I don't have the DTMP present to be able to incorporate into the DNA as a nucleotide. And what happens to DNA synthesis? I decrease DNA synthesis. So with this reaction that we kind of talked about here, it's really, really crucial to remember that if you're inhibiting this enzyme, thymidylate synthase, it's 5-FU. If you're inhibiting this enzyme in this similar process, dihydrofolate reductase, it's methotrexate. Either way, both of them are shutting down thymidine synthesis, and that's shutting down DNA synthesis. This is an important thing to remember because oftentimes we use drugs like leucovorin or folinic acid, and that actually it's interesting because it can be a rescue drug in methotrexate, but enhance the effect in 5-FU. And that's just a little kind of thing to add on there to note. But again, difference in the mechanism is dependent upon the enzyme, but the end result is the same. All right, cytarabine and gemcitabine are kind of cool. When we talk about these, what happens with these, and we'll group them together. So cytarabine and then gemcitabine. What happens with these is that these kind of basically, when you ingest them, they're called nucleoside analogs. Nucleosides are basically, you have a ribose sugar and you have the nitrogenous base, but you don't have a phosphate group. And so that's how you ingest them. Once they get into the body, they get phosphorylated, and then they generate kind of things that look like nucleotides. And so that's what happens when they get into your actual cells. they get brought into the cell, and I don't want you to know these. If you're really hardcore in biochemistry, yes, cytarabine technically gets broken into what's called ara-CTP. Gemcitabine technically gets broken into something called DF-DCTP. All I want you to know is that they are nucleoside analogs, right? And so they're going to be nucleoside analogs. All that means is they are going to have the ribose sugar, they're going to have the nitrogenous base, they're not going to have the phosphate group that gets added on. And they make these things that look like nucleotides, dude. It looks like a nucleotide. And so what happens is, here's this enzyme, DNA polymerase. DNA polymerase is moving down, it's reading the nucleotides. All right, it's A, all right, cool, let me add on a T. Here's a G, oh, let me add on a C. And then what it does is it grabs in its back pocket, and it grabs these, and these tend to look like cytosine or cytidine triphosphates. That's kind of what they look like. So they're CTP, let's say, lookalikes, or we'll call them mimics, essentially. I think that's the best way of saying it, is that they're essentially CTP mimics. All right, so cytidine triphosphate mimics. So what happens is, whenever the DNA polymerase goes in its back pocket to grab a cytidine, because it's got a guanine over here that it wants to pair with, it adds this one on. And when it adds it on, there's no point where the next nucleotide can attach. And so it terminates the DNA elongation process. And so when you incorporate these in, essentially what it does is, it inhibits DNA elongation. That's really what I want you to think about. It inhibits DNA elongation. So it's kind of like a termination point, elongation. The other thing is that some of these analogs, if you have too many of them, DNA polymerase is regulated by the number of like nucleotides that it has around it. So if you have enough of these, guess what they could do? They can inhibit the DNA polymerase, bro. So tell me that's not cool too. So they can inhibit the DNA polymerase. If I inhibit DNA polymerase, doesn't matter if I have nucleotides present, I won't be able to elongate that process. So again, when these get into the actual cell, they get phosphorylated. They look like cytidine triphosphate. Here's the DNA polymerase, right? It can be inhibited by these, but on top of it, let's say it's not inhibited. It's reading, and it's adding on the complementary base. But then let's say it gets to this one, you add on, let's just keep it going. Let's say, oh, I got the blue one, I'm gonna add the red. Okay, blue one, I'm gonna add the red. And then now it grabs on, and it grabs one of these CTP mimics, and it adds this one on. That's where it ends. It terminates right there. You can't add any more, so you don't have the next thing to add, and that kind of inhibits that DNA process from elongating, and it terminates the DNA replication process. So that's what I want you to remember for these guys. Now, gemcitabine is interesting because at high concentrations, it can even inhibit another process. There's another enzyme we talk about up there. It's called ribonucleotide reductase. So ribonucleotide reductase is an enzyme that converts a ribonucleotide into a deoxyribonucleotide. And gemcitabine at high concentrations can inhibit that enzyme. All right? That's the pyrimidine antagonist. What about the purine antagonist? So for the purine antagonist, this is your double ring structures. And so this is going to be that 6 + 5. This is going to be things like the adenine, the guanine, right? With these, when we talk about things that are acting like that, or mimicking those types of triphosphates essentially, we have things like 6-mercaptopurine, azathioprine. This is a really powerful drug category. And these also can act as immunosuppressants as well. And then we'll add this next one in, and these are commonly used as well, but this is called cladribine and fludarabine. And these are interesting because they really work really well in certain types of leukemia, like hairy cell leukemia and ALL. So really, really interesting drugs here. I think the reason why is if you come across this in your kind of like your textbooks, especially like the step one, I'd like to talk about this. There's something called adenosine deaminase. And adenosine deaminase is really interesting because it's present in lymphocytes. And sometimes patients can have resistance to adenosine deaminase. These two drugs have ADA resistance. So they don't get affected by adenosine deaminase. And because of that, they can still produce a pretty profound effect in lymphocytes and kind of prevent their proliferation. So that's what's important about these, is that these are ADA resistant, adenosine deaminase resistant. All right. Let's talk about this one here. So azathioprine is essentially kind of like, it's the prodrug of 6-mercaptopurine. So when you give a drug like azathioprine, right, let's say that you give a drug like azathioprine, you ingest it, it gets taken into the body, it actually gets broken down into 6-mercaptopurine. So that's really what, they're kind of the same thing. When you ingest this, it gets broken down to 6-mercaptopurine. When 6-mercaptopurine gets into the body, what happens is it gets acted on by an enzyme called HGPRT. All right, so there's an enzyme here called HGPRT. And that HGPRT is going to convert 6-mercaptopurine into what's called 6-thio-IMP. 6-thio-IMP is like a weird little son of a gun. It's basically kind of a molecule that can shut down so many different types of pathways. It's kind of crazy, to be honest with you. It can shut down essentially a couple different things. One is it's going to decrease your PRPP synthetase. You know there's an enzyme that helps you to take PRPP and glycine and make something called IMP? That's one thing. Another thing is it also inhibits another enzyme called adenosine deaminase. And that's basically responsible for converting adenosine and deoxyadenosine into IMP or deoxy-IMP, all right? So essentially what's gonna happen is it's going to decrease a molecule called IMP. IMP is important. And the reason why IMP is important is because IMP is basically a precursor for making something called AMP and GMP, right? Which is your adenine monophosphate, your guanine monophosphate, which eventually these need to get turned into deoxynucleotides and then incorporated into the DNA. So eventually they're converted into deoxynucleotides, but this is supposed to make these. So if you give this drug, it inhibits enzymes that help to make IMP. So if you drop the IMP levels, you drop the AMP, you drop the GMP. And if you drop these levels, guess what? You drop the synthesis of DNA because you need these. Once they get converted into the deoxynucleotide form, you need these to make DNA. That's one of the things I want you guys to understand here. All right. The other thing here is that these can also, this thio-IMP can act as a nucleoside analog. Right? The same thing like this. So it can act like a mimic. Basically, it can act as we call this a nucleoside analog. And these are basically mimics of certain types of triphosphates or deoxynucleotides. So that's one of the big things here is that it's also going to act as a nucleoside analog, which is going to shut down the DNA elongation and maybe even inhibit, what's this beautiful blue enzyme here? DNA polymerase. All right, so as the thioprine 6-mercaptopurine gets taken into by the body, converted into 6-thio-IMP. 6-thio-IMP works on a couple different enzymes, adenosine deaminase, PRPP synthetase, adenylosuccinate, dehydrogenase, and decreases IMP levels. Less IMP means less of these eventual deoxynucleotides to make DNA, and also can act as a nucleoside analog to get incorporated into the DNA and terminate DNA elongation. Now, cladribine and fludarabine are nucleoside analogs, just like this kind of 6-thio-IMP. So when we talk about cladribine and fludarabine, what happens with these puppies? Fludarabine. Oh, you've just made it, guys. These get brought into the cell, and then what happens is they act like nucleoside analogs. They're basically a nitrogenous base with a ribose sugar. They don't have a phosphate. They get into the body, they get phosphorylated, and they turn into kind of a deoxyribonucleotide. But they are not a true nucleotide. They're a mimic. And usually for these, it's going to look like an ATP mimic. So it's going to look like an adenine triphosphate mimic. All right, usually this is going to be something like, for example, this is ERA ATP, and this one can also make kind of an ATP mimic as well. Basically, they're going to act like a nucleotide. DNA polymerase will be none the wiser. It's going to run down, it's going to say, okay, there's a red one, I'm going to do a blue one. There's a red one, I'm going to do a blue one. There's a blue one, I'm going to do a red one. There's another blue one, I'm going to do a red one. And then it's going to get to this red one, it's going to say, oh, I need a nucleotide, and then it's going to grab this ATP mimic. When it grabs the ATP mimic, it'll bind it on. But guess what? There's no point where you can connect the nucleotide on. And so what happens? In the same way, we terminate DNA elongation. That's the concept here. And also these in high amounts, what can they do? they act on another another receptor site or a site on the DNA polymerase, bind onto it, change its shape, and so now it can't actually do its function. So it can also inhibit DNA polymerases, right? So that's the concept here, is that these are all going to get incorporated here. And what are they going to do? They're going to, same thing, inhibit DNA elongation. They're going to terminate that process. All right, wow. Now, with all of this being said, there's one more thing I want to add up here, all right? The reason why I say that is when we talk about these, we see these mechanisms, these three primary mechanisms. There's one more thing that I want to discuss. When we talk about purines and we want to turn it into a ribonucleotide, right? All we're doing is we're adding on a sugar and a phosphate group. But then when I want to take this ribonucleotide and turn it into a deoxy one, so I can actually incorporate it, I can't incorporate this into DNA, I can only incorporate this into DNA. I need an enzyme. And this enzyme is going to work right here. And this enzyme is called, I'm going to abbreviate it for right now, but it's called ribonucleotide reductase. All right? So we need to have drugs. We actually have drugs called ribonucleotide reductase inhibitors. We don't talk about these much because it's primarily like if we had a pure drug that does this, it's only truly one. It's really only one drug that truly comes in here and actually inhibits this enzyme. And if I inhibit this enzyme, can I turn this from a ribo or an oxy to a deoxynucleotide? No. And so I decrease my number of deoxynucleotides. I decrease the DNA process. Tell me that's not cool, right? So what would this drug primarily be? Well, I told you, gemcitabine has ribonucleotide reductase activity at high concentrations, but it's not a pure RNR inhibitor. The primary one is called hydroxyurea. And we talk about this in things like sickle cell. We talk about this in myeloproliferative neoplasia, and that's where it really comes into play, because it's actually going to help to decrease the deoxyribonucleotide processes, decrease the DNA formation in these rapidly proliferating stem cells inside the body. All right. We hit pretty good on the antimetabolites. Let's now move on to the next case here, which is, okay, we got to keep going down the line. We hit the S-phase inhibitors. Let's move into the next part here. We did antimetabolites. Let's talk about topoisomerase inhibitors. All right, so now we're going to move on to the next category, which is our topoisomerase inhibitors. So again, it's kind of like in the general discussion of what topoisomerases do. We talked about this a lot in the DNA replication video, but if you guys remember, let's say that here we have our replication bubble, right? And then we create something called the replication fork. Because you know, there's going to be that enzyme helicase, which is just going to be barreling through, unwinding the DNA and creating this little replication fork. And this area where it's kind of all occurring is called the replication bubble. Well, what happens is whenever helicase is kind of unwinding, proximal to that, it gets really, really tight. The DNA gets supercoils. And so you have these enzymes called topoisomerases, right? So this one right here is going to be topoisomerase, and we're going to talk about this one, type 1, right? This one over here we'll talk about in a second is going to be topoisomerase 2. Well, what happens is topoisomerase 1, let's just say we kind of show this with two little arms. One arm is going to make a cut, all right? So basically, let's say here's all this like really, really tightly wound up DNA. Imagine this. If this guy is trying to unwind the DNA and it hits into this area where it's like super densely coiled, is it going to be able to keep unwinding? No, it's going to hit like a road block. So what we want to do is we want to unwind the DNA just a little bit to reduce the supercoil so helicase can continue at that replication fork. So topoisomerase does this one arm that makes a cut, lets it unwind. After it unwinds, then it takes its other arm and says, okay, we unwound it pretty good, we got rid of the supercoil, boom, let's click that strand back together. And that's what it does. And it has that other arm, which is going to religate them. You can kind of think about it like this. Here's the supercoiled DNA. Here's your supercoil, all right? The supercoiled, let's say supercoiled DNA. Then the topoisomerase is going to come in here. What's it going to do? It's going to make a cut, right? And so when it makes that cut, it introduces this little cut right here, right? There's the cut. Then what it does is, when it allows that cut to take place, it allows this thing to kind of uncoil a little bit. So it allows for it to unwind. So that's what's going to happen, is we're going to allow for unwinding of DNA, and that's going to help to alleviate some of these supercoils. Here's the next thing, is then what we want to do is I'll represent this in a different color here. We'll do blue. Once we've allowed, you see how it's really, really tightly packed here and it's less tightly packed here? Then what we'll do is we'll bring these together. We'll religate them together, right? So then we'll have religation of unwinded DNA. When we give topoisomerase inhibitors, what we want to do is we want to allow for that arm to cut, to go ahead. But when we want to glue it back together, we want to block that one. So when I give a drug like a topoisomerase 1 inhibitor, there's a couple different drugs. Now I remember the one, it looks like an eye. This is the only I remember the different types of drug categories is irinotecan. So it looks like a 1. So irinotecan and topotecan. These two drugs are going to be topoisomerase 1 inhibitors. What they're going to do is they're going to come over here and they're going to inhibit this enzyme. If I inhibit this enzyme, what will happen is I'll allow for the supercoiled DNA to make a nick. So this is going to occur, and I'm going to let it unwind. What I'm going to do is whenever it comes time to religating it, I'm going to inhibit that step. Now, this will stay broken. So it's going to cause this single-stranded DNA break. And we'll kind of represent this as like a little jaggedy edge here and a little jaggedy edge here. We're going to cause single-strand DNA breaks and we're not going to religate them. So here's going to be a single-strand DNA break right here. And then what we're going to do is we're not going to allow for the religation to occur. So then if you think about that, what that'll do is that'll allow for this thing to crazy unwind. And also it'll still create torsional stress proximal to that replication fork. So helicase still won't be able to do this. And on top of that, this DNA is going to unwind so significantly, you're not going to allow for DNA replication. So that's what I want you guys to think about. Supercoiled DNA, we will make the cut. So there'll be a cut, single strand DNA, cut, and unwinding of the DNA. What we will then not allow for this enzyme to do then is to religate it. It'll stay broken, unwind, and DNA replication won't be able to occur. That should make sense. Topoisomerase 2, it's a similar thing. It's going to inhibit topoisomerase 2. Now, one is it causes one single-stranded break. Here's your supercoiled DNA. Right? What's it going to do? It's going to boom. It's going to break both strands. So it's going to cut double-stranded DNA, and it's going to allow unwinding of the DNA, the supercoiled DNA. All right, unwinding of DNA. So we're going to make the cut, then we're going to allow for this thing to uncoil. But then when it comes back to religating it, are we going to allow for that religation to occur between the two double-stranded DNA strands? No. And so that's what we're going to prevent is we're going to not allow for the religation of the unwinded DNA. All right? So what's going to happen is when we give these drugs, topoisomerase 2 inhibitors, what are they going to be? This is etoposide and teniposide. These drugs are going to work to inhibit, again, the topoisomerase inhibitor. And again, it's got the two arms. It's got the lyase domain where it's going to cut, and then it's also got the actual ligating domain where it's going to religate them together. This is going to, again, inhibit that religating domain. And so this thing will cut the actual double-stranded DNA. And I remember that. One is it makes a single or one strand break. Two, it makes a two-strand break, or it breaks both strands of the DNA. The only thing that's going to be different here is what's going to happen. Here's my double-stranded DNA break. Am I going to religate this puppy on these jaggedy edges? No. And if I don't allow for that process to occur, I'm going to, again, allow for the DNA to unwind. And not only that, but then I'm going to continue to have all of this torsional stress proximal to where the helicase is working on the replication fork. So it's going to hit a roadblock, and then all of the DNA downstream is going to unwind, and you're not going to allow for proper DNA replication. That's the key here, okay? So that's our topoisomerase inhibitors. So far, we take our nucleotides, our nitrogenous bases, we make nucleotides. Nucleotides get incorporated into the DNA. Antimetabolites are inhibiting the nucleotides forming DNA. This, we already have DNA forming, and we're trying to replicate it, but we're going to prevent that from continuing by targeting the actual enzymes that regulate supercoils. All right, the next step. So this is, again, going to be the topoisomerase inhibitors. Now we're going to come down and talk about microtubule inhibitors. Microtubule inhibitors can be broken down into two categories. So polymerization inhibitors and depolymerization inhibitors. So your polymerization inhibitors, I like to remember the vinca alkaloids. All right, so the vinca alkaloids. And there is two categories here, or two drugs that I want you to remember. And it starts with a V. So it's vincristine and vinblastine. So vincristine and vinblastine. All right. What happens is these drugs are really cool. So you have these different types of tubulin. So you have what's called alpha tubulin, and then you have what's called beta tubulin. And then when these two combine, they make heterodimers. And these heterodimers is going to be your tubulin dimer. So we'll have what's called a tubulin. It's a heterotubulin dimer. And then if you take a bunch of these dimers, guess what? And you combine them together, you will make your microtubule, right? This is how you make your microtubule. Now, microtubules are an extremely important component of allowing for connection between what? Between the centrosome, which is kind of like this area here towards the end where the mitotic spindle begins to form. This is going to send these microtubules out from the end poles here. So here, what we're going to do is we're going to have this centrosome here towards the other end. What we want to do is we want to be able to start sending these little pieces out. We want to send out these mitotic spindles, which are pretty much primarily microtubules. In order for me to do that, to allow for that process to occur, I have to cause these things to be formed. I have to polymerize them. So I need to go from here to here to here. And then I need to allow for this one to go from here to here. This one from here to here, here to here, right? So I start off with the formation of the spindles, right? Of the spindles. And then from here, if we move down, look what happened. We kind of just started to kind of inch them out. Then what we're going to do is we're going to go to this next stage. And at this stage, this is what we call metaphase. This is basically whenever the microtubules meet the chromosome at the kinetochore, right? Right at that centromere, there's a little protein called the kinetochore. We're connecting those microtubules at that portion there, at that centromere where the kinetochore protein is. Then what we do is in order to separate, so here's the chromosome at the centromere, we can split these into sister chromatids. But in order to do that, I need to break the microtubules down. I need to depolymerize them because they were really long, connecting all the way to the equatorial plate. Then what I need to do is I need to shorten them to bring this sister chromatid which way? I want to move this one this way, this one this way, this one this way, this one this way. So I need to depolymerize them, right? And this is going to be the anaphase. So polymerization of microtubules really influences metaphase. So if I give a drug like these, the vinca alkaloids, what they do is they come over here and they bind to the beta tubulin. When they bind to the beta tubulin, basically they inhibit the formation of dimers. And when you inhibit the dimer formation, you get less of these, and then you inhibit the formation of microtubules. So you inhibit the polymerization. So at which point are you going to inhibit? You're not going to allow for this metaphase portion. So this gets shut down. I can't bring these out and connect to the actual chromosomes. And therefore, if I can't connect to them, would I even be able to proceed to anaphase and separating the chromatids into each cell? So would I be able to take one cell and prevent it from forming two cells? Yes. Do you see how this is an M phase inhibitor? But it's particularly at the metaphase portion. So we've talked about drugs like the Antimetabolites, how they work in the S phase. We talked about the topoisomerase inhibitors, how they inhibit DNA replication by working at the supercoils. Topo 1 is S phase, Topo 2 is G2 phase. Which ones are these doing? This is working in mitosis, particularly at metaphase. Compare over here now. If you compare over here, now we're going to go the opposite process. So depolymerization inhibitors are called taxanes. So these are your taxanes. And this is going to be two drugs. We're going to have what's called docetaxel. So we have what's called docetaxel and paclitaxel. And we're going to start off with what? We're going to start off with a microtubule. And then basically we're going to go the opposite direction. We want to take the microtubule and we want to break it down into our dimers. So we're going to break it down into our tubulin heterodimers. So this is going to be our tubulin dimers. And then we want to break these down into your alpha tubulin and your beta tubulin. Because what we want to do is we want to start breaking these microtubules down into their small components so that way we can start moving what? So think about this. Here we start off, we have the mitotic spindle beginning to form. Then they move and they connect to the chromosome at the centromere via the protein, the kinetochore. Then, and this is via the polymerization. Then we need depolymerization to start breaking down the microtubules and shortening them so that they can move in which direction? They can move towards this pole and this pole, because I want to eventually have two cells with the same amount of genetic material. So this portion here, which I want them to depolymerize, right? This is where I'm inhibiting it, right? So we start off with the mitotic spindle. It's beginning, right? So mitotic spindle is forming here. Then we have our metaphase kind of like right here where they're connecting along the equatorial plate. And then we have anaphase. At which point is depolymerization supercritical? If we think about this pretty straightforward, well, it's not this one. It's really primarily this state. And so what we're going to do is we're going to inhibit right here. We're not going to be able to separate the sister chromatids, and therefore you're not going to get enough genetic material into the appropriate daughter cells. So this is what I want you guys to remember, is we're inhibiting the depolymerization. So you're getting less tubulin dimers, you're getting less of these alpha tubulins, less of these beta tubulins, and you're getting these microtubules to stay in their stabilized state. They're staying in the stabilized state and they're not breaking down. All right, so far, we've talked about S phase inhibitors with antimetabolites. We talked about the G2 phase inhibitors, which is going to be particularly the topoisomerase 2. We talked about the M phase inhibitors, which is going to be your microtubule inhibitors. Let's talk now about some of those cell cycle non-specific ones, the ones that are going to be able to inhibit any phase. It's not a specific phase. They could do G0, G1, G2, S phase. It doesn't matter. They could do the G0 phase if they wanted to. These are going to be your alkylating agents and your antitumor antibiotics. Let's move on to those. Antitumor antibiotics are actually really interesting and they're extremely potent, especially the cell cycle non-specific ones. The cell cycle specific is your bleomycin. And again, this is like a broken record, but it's really going to be specific in which phase? So it's the G2 phase. All the other ones that we're going to talk about here, they're going to be more particular towards, again, non-specific. So it can occur at any state. When we talk about bleomycin, what's really interesting about bleomycin is, let's say here's bleomycin. It can have, I'd say, an interesting kind of thing. He can have a DNA binding domain, right? So here's like a little portion where it could bind to the DNA, right? So let's say here we have a portion called the DNA binding domain. And let's say here it clicks into the DNA, right? So it's binding onto the DNA. So it has a DNA binding domain. DNA binding domain. All that means is it's the portion that binds with the DNA, right? The other portion is kind of where it's a little bit more critical. The other portion is like right here. This is an iron binding site, all right? So right here is a metal binding site, but primarily this is an iron binding site. And what happens here is whenever it binds to the DNA, the bleomycin, right? Iron can kind of stick into this pocket. When it binds into this pocket, it creates a I'd say like a complex that can generate reactive oxygen species. So essentially, it could take something like oxygen that may be nearby and convert it into different types of free radicals, right? So you can generate a lot of reactive oxygen species, essentially. So this is going to bind with the DNA, helps to bind iron. Iron is going to convert this into different types of superoxide anions, maybe make hydroxide anions, right? So you'll make the hydroxide ones as well, right? And these are going to lead to high amounts of these reactive oxygen species. Reactive oxygen species are extremely dangerous to proteins, lipids, and DNA. And guess what this is going to go and do? It's going to go act on the DNA and lead to damage to the DNA. And when I start damaging the DNA, guess what? If I don't have those repair genes that are coming in and fixing this, guess what's going to happen? This cell will not be able to go through the replication process. So it'll produce DNA damage. And the key thing is if this does not go through the actual repair process, so if our DNA repair enzymes aren't able to get in there and fix this during that G2 phase, what's going to happen? It'll proceed to go into dying. It actually will undergo death. So that's why this is extremely important. So bleomycin, very, very powerful type of drug, works in the G2 phase, has a DNA binding domain where it binds onto the DNA, has an iron binding site where it binds into this little pocket. Then it can create a complex which generates reactive oxygen species, produces DNA damage. If your DNA repair enzymes don't fix this, this will die. This cell will die. All right, similar concept. Now we have the cell cycle non-specific agents. So this is going to be things like doxorubicin, and there's another one called daunorubicin. So daunorubicin. And then there is another one, I'll briefly talk about this one, called dactinomycin. Now, what I want to do is, dactinomycin, really it only does one of the three functions that doxorubicin and daunorubicin do. I want to put a little asterisk next to this because this thing has three different functions. So what I'm going to do is I'm going to talk about the three different functions it has, and then I'll tell you which one of those three only this one has. All right, let's say here's the drug, right? It obviously has a DNA binding domain, right? That's one thing. So one of the things it can actually do here is it has a DNA binding domain. So that's really, really important. So it allows for it to be able to bind to the DNA, right? So it can stick to the DNA. The other thing is it has a iron binding site. Same thing, so it has an iron binding site. And so because of this, it has a couple different types of things that it can do. You'd probably be like, oh, it's the same thing like bleomycin. Not necessarily. It can do a couple different things. Because it has a DNA binding domain, it can bind in areas near another enzyme called topoisomerase 2. And so what this has the ability to do is it has the ability to inhibit a very beautiful enzyme that we talked about already. So if you remember that enzyme's function, you'll remember this is one of the three functions of doxorubicin and daunorubicin. This is your topo 2 enzyme. And so it's going to inhibit the topo 2 enzyme. Can you guys remember, if we have this drug bind onto the DNA, inhibit the topo 2 enzyme, what's that going to do? It's going to produce, come on, double-strand DNA breaks that don't get religated. And so essentially you end up with increasing double-stranded DNA breaks, and this leads to unwinding of DNA. And basically you can't allow for that replication fork to continue. And so this is going to lead to decreased DNA replication. So this is going to decrease the DNA replication process at that replication fork, right? It's going to cause all that torsional stress. All right, that's one of its three functions. I think you can guess the next one. If it can bind to the DNA and it has this little iron binding site, well then guess who can bind in there? Iron. And if you can bind iron, what can it do? It can take oxygen and turn this into like a superoxide anion. It can make hydroxyl anions, and these are called reactive oxygen species, and these will produce DNA breaks. And if you produce DNA breaks and you don't allow for that actual fixing, you don't allow for those DNA repair enzymes to come in and actually fix this process, then what happens? Then the actual cell will die. So that is really the key thing here, is that we're going to generate lots of reactive oxygen species, lots of DNA breaks. And that is definitely going to hinder DNA replication, because if you can't fix it, the cell will die. One other one. Doxorubicin, daunorubicin, and dactinomycin are kind of like, they're kind of cool in the sense that they can really, because they have a DNA binding domain, they can actually interact with certain nitrogenous bases. And basically they can kind of act like like a little, a person to block between the two. So if I have a nitrogenous base over here, a nitrogenous base over here, and I have this drug in between, it can intercalate. It's called intercalation between the actual DNA strands. Now imagine if I have this, intercalating these, can DNA polymerase get in here and read this? No. So what happens is, whenever DNA polymerase is trying to go down and read this DNA strands and create a new strand, it runs into this big beast. And guess what? He says, I ain't moving. And we inhibit this process. And so that is really key, is so we can have something called intercalation. So intercalation of DNA, and this inhibits DNA polymerase. So again, what happens here is this gets in and binds in between the nitrogen, and this is not a covalent bond. It's a non-covalent bond. That's what alkylating agents do, not this one. It just goes in and kind of creates a non-covalent bond, intercalates in between the DNA, blocks DNA polymerase from reading these strands and making new ones. So it's going to inhibit DNA polymerase. And if you inhibit DNA polymerase, can you replicate the DNA? No. And if you can't replicate that DNA, you ain't gonna make no new cells. And that's good with cancer, right? So that's the three mechanisms. Three mechanisms. One is it inhibits the topoisomerase 2 enzyme. Second thing is it increases reactive oxygen species. Third is it intercalates the DNA. Dactinomycin only does the third action. That's all I want you to remember. Here, I'll write that down just so that you guys know that. This has three. Dactinomycin really only does intercalation. So it really only performs intercalation of DNA. Whereas doxorubicin and daunorubicin, this one, it can do it all. It can do all three. So that's what I want you guys to remember. So far, with anti-tumor antibiotics, we have cell cycle specific, bleomycin, G2 phase, only does reactive oxygen species. Cell cycle non-specific, doxorubicin, daunorubicin, three types of mechanisms. Reactive oxygen species, inhibits topoisomerase 2, which we already talked about, and it kind of acts like a little blocking person, a security guard between the nucleotides. You then don't allow for DNA polymerase. It hits a roadblock. It can't continue if you have this big boofy thing blocking its way. Dactinomycin only does that little big boofing thing where it blocks the way. That's what I want you guys to remember here. All right, so let's move on to alkylating agents. Alkylating agents are cell cycle non-specific agents, meaning that they don't affect any specific phase. You know, they don't really have to work only in G1 or S phase or M phase, anything like that. So they can work at any phase, including the G0 phase. So the thing about that is when we talk about these drugs, we can break them into three different categories. One is your nitrogen mustards, right? And it's not the same mustard you put on your glizzies as at a baseball game, but there's a couple different drugs here. One is called cyclophosphamide. Another one is called I-phosphamide. And then the last one here for the nitrogen mustards, you know, we don't use this one often, but you can use it in multiple myeloma, and that's called melphalan. It's usually as a conditioning agent sometimes. Triazines, these are basically, they're all going to form generally some type of covalent bond, especially these. This one right here is going to form some type of methylation group, and this is usually going to be what's called temozolomide. And then lastly is you have your platinum compounds. Platinum compounds, basically they can generate covalent bonds between, again, the different components of the DNA strands, whether it be inter or intra, and we'll explain what that means in a second, but they utilize a metal ion that these nitrogen mustards don't, thus the name platinum. And so this is going to be things like cisplatin, carboplatin, and then you also have oxaliplatin. Now, with all of this being said, how do they work? We're not going to complicate this. It's not complicated. When you give these drugs, the nitrogen mustards, all they really do is they're basically going to be, think about it, it's like a piece of gum. Here's this molecule. And this piece of gum is going to kind of form in between the DNA strands. Now, it can do this in a way where you could actually generate bonds maybe between this nucleotide and this nucleotide. That's called a intra-strand crosslink. Or you could form covalent bonds between this nucleotide and this nucleotide. That's called interstrand crosslinks. And so you can get different types of crosslinks, right? All I want you to know is that you're going to make a lot of crosslinks between the DNA. If I make crosslinks between the DNA, and these are covalent bonds, so let's call these crosslinks, and then let's annotate something here. This is, again, what kind of bond? We'll just represent this in green over here. These are going to be very powerful types of bonds, strong bonds. This is a covalent bond. All right, so this is a covalent bond. The ones up here that we talked about, which are again a part of your anti-tumor antibiotics, doxorubicin, daunorubicin, dactinomycin, those intercalate, but they form weak bonds. They don't form covalent bonds. And again, these crosslinks can be intrastrand or interstrand. Think about how this works. The DNA is supposed to be sandwiched together, right? So these are generally, they're forming hydrogen bonds between this nucleotide and this nucleotide, right? So they should be very, very close. This guy comes in and sandwiches between them and says, I'm going to hold you together. And now, if it holds it together, when I want to take and separate, right? I want to separate these out. And we use different types of proteins to do that, right? For example, what are the different types of protein that separates the actual DNA strands? It's called the DNA A protein. What helps it to keep it apart? Single-stranded binding proteins. What's the enzyme that comes through and opens up the replication fork? Helicase. What's the enzyme that runs through and reads the DNA and makes new DNA? DNA polymerase. Can you do any of that? No. So all of these enzymes that you need to perform these functions, for example, things like DNA polymerase, right? Here's one. He's supposed to get in there, your DNA pol. He's supposed to get in there and read the DNA. Helicase is supposed to get in there, and it's supposed to separate the actual DNA strands at the replication fork. Can he do that? No. It's literally, it's like gluing the strands together. And then there's other ones, right? So you have DNA A proteins. I like to do it like this because it kind of looks like it hooks the DNA and separates it. And you also have other ones called single-stranded binding proteins. Either way, all of these puppies are supposed to literally be utilized to get into the DNA, read it, and make new DNA. Are you going to allow for that process to occur? Are you going to allow for that separation and reading? No. All of that's inhibited. And so what you do is you decrease, essentially, DNA replication because you can't separate the strands. It's the exact same down here with this one. So that's why it's kind of nice when we think about this, because the only thing that's different, so we're going to take a little beat here. We're going to come over here for a second because it is exactly, and I mean exactly the same. These are compounds. Actually, let's do this the correct color. Come on, Zach. You're going to have this guy coming down. But what's the difference? This is going to be a piece of chewing gum, let's just say, and it's going to have a specific ion called platinum in it, all right? And what's going to happen is this is going to go in here, and this chewing gum is going to do the exact same dang thing. It's primarily more, you see this being more intrastrand, but it can cause interstrand. But either way, this may be connecting these and these. This may be connecting these and these. But either way, the same concept exists. Can I have all of these enzymes get in there and separate the DNA and read it? Can DNA polymerase get in there? No. Can helicase get in there? No. Can our DNA A proteins separate the strands, and can single-stranded binding proteins hold it apart? No. And if we can't do these particular functions, can I even allow for DNA replication to take place? No. So I'm going to increase my crosslinks, which decreases that DNA replication. That's the concept I want you to understand, right? So we're forming crosslinks, and these crosslinks, they're not weak, man. They're pretty strong. And because of that, it makes it really hard to truly perform that replication process. And that's what I want you guys to consider here, is that we are really doing it through that concept. Now, Temozolomide, similar, but it's not completely the same. For example, when we talk about temozolomide and these triazines, what happens is this drug, it gets converted into like a complex that primarily its job is to methylate. And so we call this component, it's called MTIC. Don't go too crazy memorizing all of that. What I want you to really just simply know is that what it does is it takes a methyl group and it adds it on to specific nucleotides. And when you add this methyl group on, what it may do is it may allow for a mismatching. So maybe this one was supposed to go with this one. Now it mismatches it with the improper nucleotide. If you cause mismatch kinds of processes and you don't allow for that repair process to occur, that's when we start seeing issues, because it actually does distort the DNA helix. That's where we start to see problems. And so because of this, you end up with what's called a mismatch. You end up causing mismatch nucleotides, right, through this kind of issue. And this, again, makes it really hard. It distorts the DNA double helix, but on top of that, it really makes it hard for all of these enzymes to perform the DNA replication process. And that's going to shut down that DNA replication. All right? So you're going to increase your mismatch between those nucleotides and you're going to shut down that DNA replication by methylating these. And that's going to be the issue with this drug. All right. So with alkylating agents, again, we have these three categories. The biggest thing that I need you to remember is they like to form crosslinks. The difference between these two is this has platinum involved. This one is it methylates structures to allow for that improper matching of nucleotides. All right. Now that we nailed this one and destroyed it, let's go into the next portion. Because we talked about all the drugs that influence the cell cycle. Now let's look at this in a different way. Let's say, okay, we talked about the drugs such as the antimetabolites and how those influence purine and pyrimidine synthesis and incorporating that into the DNA. We talked about topoisomerase inhibitors and how they really act on the supercoils and how they really kind of prevent the religating when we have supercoils. We talked about the microtubule inhibitors and how they basically inhibit them from polymerizing to connect at metaphase and how they inhibit them from depolymerizing to break apart during anaphase. We talked about drugs that basically can intercalate. In other words, they can get into the DNA and kind of just block it. They'll block DNA polymerase. They can generate free radicals. And then in some ways, they can even, doxorubicin and daunorubicin, generate some topoisomerase II activity. And then lastly, we talked about the drugs who can generate a cross-linkage of nucleotides with a strong bond, making it hard to separate the DNA, generate a replication bubble, fork, and then replicate the DNA in general. Now that we've done that, let's move on to what about drugs that can target upstream pathways that influence the cell cycle, because it all leads to that CDK4 and 6. What about all those different pathways that influence CDK? CDK itself, do we have drugs for? Let's talk about that. All right, so now we're going to talk about our protein kinase inhibitors. So, you know, there's a lot of different proteins that are basically regulating your cyclin-dependent kinases, right? Because that's really what we kind of talked about, that really kind of pushes the cell list. Hey, let's go through. Because it's at that G1S checkpoint, and we really want to have something that says, all right, you're good, go ahead and proceed. And if we have something that's constantly stimulating that, like a gnat at a barbecue, then it's just going to keep continuing to go through that cell proliferation process. And so it's kind of reminding ourselves that we have that CDK4 and 6 enzyme. And again, do you guys remember what it does? Well, it does what? It adds the phosphate groups onto our good old friend here, the retinoblastoma protein, right? And it's holding on to the E2F very tightly. And so when it adds the phosphate groups onto that, what does it do? It then releases E2F. And if we release E2F, that E2F is a very powerful transcription factor that is capable of doing what? Well, it's capable of getting in here, activating on specific proto-oncogenes, and if it acts on those proto-oncogenes, it's going to stimulate very specific genes that are going to enhance what type of process? The initiation of going from that G1 into the S phase. And so if I want to stimulate this process and allow for that cell to go through the proliferative phases, guess what? That's what this is going to do, and it's going to really push this to occur. And that's going to allow for the cell proliferation. So what I want to do is I have to remember that there's all of these pathways coming downstream that are influencing this, right? So for example, here is this molecule. What did we say this one was called? This is called BCR-ABL. This is a constitutive, it's basically a non-membranous tyrosine kinase. It's in the cytoplasm, and it can be activated for multiple different reasons, but one of the cool things about this is that it really helps to drive some of the similar pathways that a lot of these receptors do. We also have EGFR receptors. When EGFR is activated by a growth factor, it triggers intracellular pathways. Whenever the tyrosine kinases that are associated with ALK are activated by specific growth factors, it triggers an intracellular pathway. Whenever the jaundice kinase receptors are activated by growth factors, it triggers an intracellular pathway. What are those pathways, you ask? It can all lead to an increase in PI3K, AKT signaling, which leads to the mTOR activation. It can also increase the activity of RAS, which is a GTPase, which then leads to the activity of increased RAF, which then leads to the increased activity of MEK, and this is all part of our MAP kinase pathway. And then we can also activate signal transducers, activators of transcription. And so all of these, to a degree, have the capability of increasing these pathways. The JAK-STAT pathway, that's primarily only going to increase STAT. But all these other ones can influence it pretty much the same way. And so these are generally trying to increase the activity of these specific pathways. This goes up, this goes up. And what happens is, what do these do? These lead to a downstream effect on cyclin-dependent kinases. And if you activate the cyclin-dependent kinases, what will it do? It'll trigger this process, right? So that's all of this upstream to downstream effect. Now, think about this, all right? Think about this. If I give drugs that can act on these different levels, I could potentially shut down these pathways. So let's say I give a drug that inhibits the BCR-ABL tyrosine kinases. What would that drug be? This is usually any of the tyrosine kinase inhibitors, TKIs, which you'll see here, usually end in INIB, INIB. So this one is example imatinib. If you can't remember that, that's okay as long as you see the INIB. The EGFR, right? And so, well, actually before we do that, this is inhibiting this guy. It's going to basically bind to this ATP binding site. So it'll bind to the ATP binding site, and that will inhibit this enzyme. And that'll shut down that pathway. And if we shut down this pathway, we're going to drop all of these levels. And that's no longer going to stimulate CDK4 and 6. It's going to inhibit it. That'll shut down the phosphorylation of retinoblastoma. That'll shut down the release of E2F. If I don't have E2F, can I stimulate transcription? Can this transcription factor stimulate specific proto-oncogenes? No, it's going to inhibit them. And instead, we will put the brakes on the checkpoint and say, don't go through any more cell proliferation. Tell me that's not a cool mechanism, right? We use this a lot in CML, believe it or not. EGFR TKIs. If I give this drug, right, what it's going to do is it's going to act on the tyrosine kinase portion of this receptor, and that's going to do what? Think about it. It's going to inhibit them. If you inhibit them, you shut down this pathway, and you do this. It's the same thing. It's the same fucking thing, right? Tyrosine kinase inhibitors. We're going to inhibit the tyrosine kinase of this receptor. If we do that, we shut down this receptor signaling, and we shut down this. If we give a drug like a JAK-STAT inhibitor, it's going to act on the jaundice kinase portion of this receptor, and then do what? Shut it down. And that's going to decrease this pathway. So then what are these drugs that we can talk about? Well, this is a TKI and this is a TKI. So it's going to end in INIB. It's just they have a beginning, a different kind of prefix. Osimertinib. All right, so there is the INIB, osimertinib. ALK, this is going to be alectinib, alectinib. See the INIB? And then there's the JAK-STAT inhibitors, and this is going to be ruxolitinib, and you'll see the INIB. All right? Now, you can kind of see a pattern here, and that's pretty cool when you see these protein kinase inhibitors, the small molecule inhibitors. Now, the other concept is you see this pathway, the RAS, RAF, MEK, which are a part of that MAP kinase pathway. You have drugs that can inhibit the PI3K and the AKT and even the mTOR pathway we're not going to be seeing that too much as with respect to things for step 1, step 2, but you will potentially see some drugs that are involved in the MAP kinase, which they're basically RAF inhibitors. And so let's say that I have some drugs here that are going to have the possibility of, let's kind of do it like this. They have the capability of inhibiting these pathways. And if that happens, they shut down this kind of downstream effect. What could be some drugs here? Well, there's one particular one, and it works on the RAF. Well, we call this a BRAF inhibitor. And that BRAF inhibitor that we really are speaking of is called vemurafenib. There is other ones like MEK inhibitors, but we're not going to talk about that. But BRAF inhibitors are called vemurafenib. And I like to actually remember this one with RAF. All right, so that helps you a little bit to remember that that's kind of a RAF inhibitor, which is a part of the MAP kinase pathway. All right, the last one is, we talked about all the upstream ones, kind of leading to this kind of inhibition of CDK4 and 6. If we're using these drugs, we're inhibiting that. What if I have a drug that can go straight directly to CDK4 and 6? That'd be a CDK4/6 inhibitor. If I could inhibit that, that would be the same kind of effect, and it would be very, very potent. We do use this drug. It's very, very commonly utilized in breast cancer, usually in combination with other things like tamoxifen, or usually things with like aromatase inhibitors. But this is a pretty good one in metastatic breast cancer, all right, especially if they're usually estrogen receptor, progesterone receptor positive. But this right here would be palbociclib. This is an example one. So again, usually you're looking for the ciclib portion for the CDK4/6 inhibitors. All right? So this is what I want you guys to remember when we talk about your protein kinase inhibitors. Usually if they're TKIs, they end in inib, and then if it's a BRAF inhibitor, remember RAF and vemurafenib, and then ciclib is your CDK4/6 inhibitors. Again, you're inhibiting the upstream pathways, midstream pathway, or the cyclin-dependent kinases. But either way, you're inhibiting the protein kinases that regulate the cell cycle, and you can then shut down that checkpoint where they don't continue to go through cellular proliferation. That's cool. What if I use drugs to inhibit this same pathway from this point, but it's nothing to do with the receptors or intracellular pathways here? It has to do with blocking hormones. Would you believe me? That's okay if you don't. We're going to show you. So hormonal therapy is commonly utilized in cancers like prostate cancers. It's used a lot in breast cancer, heavily in breast cancer, especially if they're estrogen receptor, progesterone receptor positive. We utilize these. So you got to go back to kind of a little bit of the hypothalamic pituitary gonadal axis. Your hypothalamus makes something called GnRH. That tells the anterior pituitary to make something called FSH and also another molecule called LH. All right, so hypothalamus makes GnRH. GnRH tells the anterior pituitary to make FSH and LH. FSH acts on the ovaries and LH acts on the Leydig cells of the testes. And this pumps out a hormone. This pumps out a hormone. The hormone that's made from the presence of these is going to be testosterone from the testes, right? From the nudes. And then that testosterone can actually be converted into something called DHT. And that's really what is the driver of patients who have prostate cancer. And so you want to be able to shut that down, right? For females, when it acts on the ovaries, it triggers them to produce something called estrogen. And so if a patient is unfortunately producing, let's say, large amounts of estrogen or large amounts of testosterone, that can lead to some particular issues here, right? Testosterone can actually eventually, again, it gets converted into what's called DHT if you really want to, or, we call this one, there's an enzyme called 5-alpha reductase, and that eventually converts this into what's called DHT. We really wanted to remember, and this is an androgen, so it can bind onto different types of androgen receptors, essentially. But estrogen, let's think about this one for a second. Estrogen, you know what it does? Estrogen can then go over here. It can actually cross the cell membrane because it's lipid soluble, it's a steroid. And when it does that, it binds on to these different estrogen receptors that are present inside the cell. When you bind on to these estrogen receptors, you activate these, and these then lead to the stimulation of our good old friends, CDK4 and 6. And what does this do? Adds phosphate groups onto, who's this friend? Retinoblastoma. Who's it holding on to? E2F. If This gets phosphorylated. What does it do to the E2F? Spits the E2F out. If E2F is spit out and it's free, right, what does E2F do? It says, all right, let me act on these particular genes, and then it acts on these specific proto-oncogenes, stimulates them, which then does what? Drives the person to go from the G1 phase, right? So it's going to stimulate some specific type of proto-oncogene, which then stimulates to go from that G1 phase to the S phase, which is just going to push the cell to keep going through that replicative cycle. All right, that's really interesting when we think about that. The other concept here is that not only is estrogen made from the ovaries, all right, but you also have testosterone can get converted into DHT, right? There's another thing here where we can take something like an androgen and convert it into estrogen, which is kind of interesting. So you know in females as they get older, their adipose tissue tends to kind of increase. And so what happens is that's why postmenopausal women are high risk for breast cancer. They can increase the production of estrogen. I'm sorry, androgens. So your adipose tissue will produce some things like androgens. And so does your adrenal gland. So your adrenal cortex can also make androgens. Now, why is this relevant? Well, when you make androgens, there's an enzyme here. And this enzyme is called aromatase. So it's called aromatase. And what happens is aromatase essentially takes these androgens and spits them out and converts them into estrogens. And so if you have a person who has a high risk of breast cancer, right, their adipose tissue, if they're obese, they may be producing a lot of androgens. A lot of these androgens, if acted on by aromatase, produces lots of estrogen. This acts in that breast tissue and increases the risk of developing things like proliferative tissue within those mammary glands causing breast cancer. So that's what we see there. The other thing here is that androgens also, on their own, again, things like DHT or other androgens, have the capability of binding onto these androgen receptors, right? And so if they bind onto the androgen receptors, they act just as though estrogen would, which was to do what? If they're stimulated, they're going to act on the CDK4 and 6. And they're going to stimulate this puppy. And if we stimulate this puppy, right, he's going to release the E2F. And if we release the E2F, it's going to drive this cell proliferation cycle. So what if I could come up with ways to shut down this pathway and shut down that cyclin-dependent kinase pathway? I may be able to shut down that cell cycle from continuing to go. So let's do that. I'm going to give a drug, which we use a lot in prostate cancer, called Leuprolide. And this is a Leuprolide is a GnRH agonist. It's a little funky, the mechanism, but it's also kind of cool how they think about this stuff. So what it does is, let's say for a second here, just follow me for a second. Initially what it'll do is, it'll help to kind of essentially stimulate the same pathway. It's going to act like GnRH. So what it'll do is it'll kind of create an environment of where you have high GnRH. If you do that, you increase FSH, you increase LH, and that increases estrogen, testosterone. You're like, Zach, that doesn't make any sense. Hold on. When testosterone and estrogen levels go up, what that does is high testosterone and high estrogen create a negative feedback. So high test, high estrogen, basically what that does is it creates a negative feedback mechanism. And when you create this negative feedback mechanism, it essentially shuts down the GnRH pathway. Because it's like a protective thing. It says, oh, you better go down. Oh, you better go down. Oh, you better go down. And then guess what happens to the estrogen levels? They go down. What happens to the testosterone levels? They go down. What happens to the DHT levels? They go down. So that improves things in prostate cancer, right? And also Leuprolide, I mean, you theoretically could drop this in patients who have maybe a need to drop those estrogen levels. Now, that's what I want you to think about is that GnRH agonists, they technically lead to, if you really want to think about it, they theoretically increase estrogen, they increase testosterone, which creates a negative feedback mechanism to shut down that pathway. GnRH agonists, they do this directly. They directly lower the GnRH effect. That drops the FSH, that drops the LH, that drops the estrogen, that drops the testosterone, the DHT. This is a drug called Degarelix. All right, now let's keep going. What if I had a drug where it didn't affect any of the FSH, it didn't affect any of the LH, estrogen got produced, but I wanted to block estrogen from binding onto the receptors? I could do that with SERMs. So SERMs basically are going to be able to come in and basically block estrogen from binding to this receptor. So when it does that, it shuts down this pathway. If it does that, it inhibits the CDK4 and 6. That then inhibits the phosphorylation. That drops the free E2F levels. That then does not activate the proto-oncogenes, and that shuts down this pathway. That's pretty cool, right? And so the drug categories here that we utilize often in ER/PR positive breast cancer is tamoxifen. And there's another drug that we can use as a chemopreventative agent called raloxifene. It's the same kind of drug category as well, all right? But that's what we would do there. Now let's say that, okay, we have another issue. We want to basically use the way of being able to block androgens from being converted into estrogen. I can do that with this enzyme. And so, or inhibiting that enzyme. And so I can use a drug to basically inhibit aromatase. If I inhibit aromatase, I basically don't convert androgens into estrogen. What happens to the estrogen levels? They fall. And then this pathway is inhibited. What would be some examples here? We use this a lot in, again, breast cancer. You can also use this, there's many other conditions, but I think the big thing to remember, we'll talk about it a lot in the indication section, is going to be breast cancer. But this is going to be things like letrozole and anastrozole. Like, these are pretty common ones that we talk about there. All right, so they're going to inhibit that pathway. The last thing is, what if I wanted to block the androgens directly? I could do that, especially in prostate cancer. I could block this directly. I could give a drug that basically comes here and says, hey, let me block these androgens from being able to bind to these receptors. And if I do that, I inhibit the CDK4 and 6, right? So that would inhibit this process, right? And that's gonna still shut down this whole pathway. What would be some drugs that I could use here? This could be something like enzalutamide. Enzalutamide. So again, when we talk about this, we can see that you can have a lot of upstream pathways that influence CDK4 and CDK6. It could be things like protein kinases, and you could have things like hormones that are influencing a lot of these. If we could target these hormones, target these protein kinases, we could potentially reduce the cell proliferative process and then treat these patients' underlying cancers. Tell me that doesn't make sense and is not cool. I hope that you guys understood this, because now what we got to do is we got to keep building. There's one more way that we can inhibit this. We can inhibit this another thing where we can again target receptors, but we don't target the kinases, we don't target the hormones. We target the receptors by using monoclonal antibodies. Let's talk about that next. All right, so let's talk about monoclonal antibodies. So these ones are going to be very interesting. Usually anytime you hear monoclonal antibody or an immune checkpoint inhibitor, you can think of the ending MAB, right? Just like with the protein kinase inhibitors, we talked about those. For the most part, like, your TKIs were ending in the INIB, your RAFinib was going to be for your BRAF inhibitors. And so that's going to be really important. For monoclonal antibodies, immune checkpoint, they usually end in MAB, and you'll see that as we go through it. But again, it's the same concept, man. We're targeting upstream pathways. And so you kind of just go back to this and it's going to become so redundant, but you just won't forget it. That CDK4/6 is really important here. And what is it doing? It's again phosphorylating which particular molecule? It's phosphorylating the retinoblastoma protein, who holds the E2F. If we add phosphates onto this, what does it do? It spits the E2F out. There's my green marker, my friends. So if we spit the E2F out and it's free, we know that E2F is a transcription factor, a pretty powerful one at that, activates proto-oncogenes, and if it activates the proto-oncogenes, what will that do? That will then stimulate the conversion, or that kind of trigger, if you will, to go from the G1 phase and say, all right, you're good, go ahead, hike, go into the S phase and begin the DNA replication process. So it'll stimulate the particular proto-oncogenes and then drive them to go through that cell cycle, right? So we have a couple other pathways that are influencing this one. You're probably like, dude, this sounds familiar, EGFR. Yeah, but we're not targeting the tyrosine kinase part of the receptor. We're actually targeting the receptor itself, the part that binds the ligands. And so I can use a drug, it's called cetuximab. So we use this a lot, particularly in colorectal cancer. And so whenever we use this one, what it will do is it will come and literally, I'm not kidding, it's gonna be a monoclonal antibody. And so you have a couple different options here. Think about this. We have here the monoclonal antibodies. And here, is going to be the growth factors. And what your growth factors want to do, the epidermal growth factors, is they want to be able to bind here. So what this monoclonal antibody will do is it'll basically kind of essentially knock these out of the way. So we're going to push these little epidermal growth factors out of the way, and these monoclonal antibodies will bind. They'll literally bind to these receptors. And when you do that, you essentially kind of inhibit them. And if you inhibit this interaction now, you're going to do what? You're going to shut down the intracellular pathways. What are those pathways? I'm so glad you asked. The PI3K, AKT, the RAS, RAF, MEK, the STAT pathways. And so all of these will be shut down. You'll shut down that PI3K, AKT, TOR pathway. You'll shut down the RAS, the RAF, the MEK, which is a part of the MAP kinase pathway, and then the STAT pathway. And by doing that, what do? Well, you then say, if I don't have these all kind of activated, what's it going to do to the CDK4 and 6? It's not going to stimulate this, therefore it will inhibit it. Are you going to phosphorylate retinoblastoma? No. Are you going to release E2F? No. Your E2F levels drop. Can you actually activate proto-oncogenes? No. Can you then put this cell into the proliferative stage? No. That is what I want you to know about this one. Another one is called a HER2 blocker. HER2 blockers, you want to think about something called trastuzumab. Trastuzumab. This one is very, very, very important one in breast cancer. And what happens here, again, it's a monoclonal antibody. So basically, you're going to have some type of ligand that will bind to the HER2 receptor. Usually these are some type of growth factor of some sort, right? And you're going to have these monoclonal antibodies. And these monoclonal antibodies essentially are going to come and block that growth factor. And when it does that, it then says, all right, cool, I'm going to shut this pathway down. And so now, when I do that, I can't trigger these intracellular signals. And if I can't trigger these intracellular signals, can that then lead to the activation of CDK4 and 6? No. And what do I do to this pathway? I shut that pathway down. So that's what I want you to remember. And then the big thing to take away from this is look at these kind of names. MAB, that's a monoclonal antibody. And so that's what I want you to think about. So we talked about drugs that target the tyrosine kinases, the INIBs. It could be the BCR-ABL one, like the imatinib. It could be the EGFR tyrosine kinase, like osimertinib. It could be the ALK tyrosine kinases, like alectinib. You have ruxolitinib for the JAK-STAT pathway. We talked about the ones that are inhibiting the RAS-RAF pathway, which is going to be the MAP kinase inhibitors, like vemurafenib. And then we have the ones that inhibit the CDK4/6, the palbociclib. We also have drugs that can block hormones that influence this pathway. And then we have monoclonal antibodies that can block this pathway. That should really give us an overall look at how we can use drugs to alter cell cycle regulation. What about this weird little thing that I want to talk about here, where it doesn't really influence the cell cycle regulation, but it does alter our immune processes, and it does alter the angiogenic processes. All right, we don't want cancer cells to be able to evade our immune system. We don't want that. And so here we're going to take, for example, let's say that we have a lymphocyte, right? So let's say it's a cancerous lymphocyte. Let's say that this is a cancerous lymphocyte, all right? And usually you'd see this in, we'll just use this as an example. This would be like non-Hodgkin's lymphoma, because in Hodgkin's lymphoma, they don't express the CD20. But here we're going to have this molecule, and this is called a CD20, so cluster differentiation. It's going to be expressed on these normal B lymphocytes and then also malignant B lymphocytes that are the non-Hodgkin lymphoma type, not the Hodgkin with Reed-Sternberg cells. All right, those are usually CD15-30 positive, CD20-45 negative, if you guys remember. But CD20 blockers, you're going to be using things like rituximab. So rituximab is a monoclonal antibody. And so if I give this drug, basically, what it'll do is it will bind to that CD20. And when it binds to the CD20, here we go. Boom, watch this, ready? We're gonna bind this puppy to the CD20. A couple different things can happen, right? One is I'm tagging it. And when I tag this, I can do a couple things. Let's say I tag this CD20 with this monoclonal antibody. And when I do that, I open up an opportunity for specific proteins to kind of bind to it. You know, there's these proteins called complement proteins. And when they see these antibodies, they love to bind onto it. And when they do that, usually it creates like this little like thing called a membrane attack complex, which is this protein pore. It gets inserted into the membrane. And then what happens is water will be allowed to influx in. And then what happens is this causes lysis. This literally causes the lysis of these cells. This kind of mechanism is usually called some degree of like opsonization. So it's usually some type of opsonization or formation of a membrane attack complex. That's one way that we can kill these cancerous cells, is we can have antibodies tag them and trigger them to undergo complement activation. Right? So opsonization would lead to complement activation, membrane attack complex formation, and you know what else? When you have these antibodies tagging this and then complements come and bind to it, it causes macrophages to get really, really like, they're like, oh my gosh. It's like you take bread and it's just bread. It's not that appetizing. You take bread and you slather some peanut butter and jelly. That's what the antibodies do. They create like a peanut butter jelly film, which now when the complements bind, macrophages want to eat it. So it enhances phagocytosis and leads to membrane attack complexes. That's what this thing's called here. So you see this right here? This is called a membrane attack complex. We'll kind of represent that here, this little channel protein. The other thing that can happen is when this binds, it can trigger kind of like intracellular pathways that activate different types of like pro-apoptotic genes, things like the Bax genes. And when that happens, it basically, I'm not kidding, it just triggers apoptosis of these cells. So they start dying. All right, so also that's one kind of direct mechanism, is it can activate pro-apoptotic genes and just initiate apoptosis in general. Another thing that can happen is that whenever we do this and we coat this, it can activate other types of killing cells. And these are called natural killer cells. And natural killer cells, when they kind of bind this, it activates them. And when natural killer cells are activated, they're nasty little buggers. And they release things like granzymes, and these granzymes are like, ooh, daddy, I'm gonna jack this cell up. And these granzymes get over here to this cancerous cell, trigger an intracellular cascade, activate those pro-apoptotic genes, and guess what? Boom, this thing gonna be dead. So dead as a doorknob now, unfortunately. This process here, which leads to apoptosis, but via indirect mechanism, is called antibody-dependent cellular cytotoxicity. Heck of a name. But there's 3 downstream pathways that rituximab triggers. Opsonization that leads to complement activation, apoptosis directly, or antibody-dependent cellular cytotoxicity. This is all going to lead to the death of these cancer cells. And usually which one? Lymphocytes. And so we use this one a lot. We'll talk about it later in non-Hodgkin's lymphoma. Now, that's one way that we can activate our immune system. The other thing that we can do, a little off topic, is we can alter the angiogenic mechanisms. You know there's a drug, it's called bevacizumab, or bevacizumab actually, I think they pronounce it. What happens with this is, again, it's a monoclonal antibody. You know these cancer cells, they're sneaky sons of guns, man. And they release something called VEGF, vascular endothelial growth factor. And they be pumping this thing out there. When that gets released, what VEGF does is it binds onto the VEGF receptors. And when it binds onto the VEGF receptors on these endothelial cells, it causes them to want to grow. And so I'm not kidding, it causes the endothelial cells to grow. And they start sprouting off these like different kind of like vessels off of them. And that's called angiogenesis. All right, we talked about that in the beginning of the video, which was about like 6 hours ago at this point, right? All right. So now, when we talk about this, here's what I want you to think about. If I give a drug like bevacizumab, that is a monoclonal antibody. And what it'll do is it'll literally bind to VEGF, right? So when I give this VEGF blocker, it is a monoclonal antibody. And so that monoclonal antibody will then bind with VEGF. When it binds with VEGF, can you then allow for this to connect, the VEGF to connect with the VEGF receptor? No. Does this interaction take place? No. Can you tell the endothelial cells to start replicating and making more vessels? No. Can you deliver more oxygen to the cancer cells? No. So then all these cancer cells, they're depending on this increased blood flow. You're starving them, which is awesome. You starve them puppies. And that is what we want, is we want to reduce the oxygen supply to the cancer cells and starve them, essentially starve them of oxygen, which can reduce their growth. So that sometimes we'll give bevacizumab. We can actually give this one actually This one's given in colorectal cancer. So sometimes if we can't give cetuximab, we'll actually give bevacizumab, all right? All right, that covers these mechanisms, which we're really overviewing with kind of the immune kind of evasion process, the angiogenic one, but this one really we've repeated it multiple different times through the cell cycle regulation. Let's finish up the last part, which kind of touches on this immune evasion process and how we can use drugs to really add our benefit of using our T cells to kill these cancer cells with the immune checkpoint inhibitors. All right, so let's move on to immune checkpoint inhibitors. So these are, again, monoclonal antibodies. Now, what's interesting about these is that they're really working to particularly, I'd say, turn the T cell on, get it to be activated. Because if you guys remember, we talked about how these cancer cells, they can increase the expression of like their PD-L1, they can cause the tumor cells to increase their expression of CTLA-4. What if I could come up with drugs to target things like PD-L1 and PD-1 and CTLA-4? That would be a true benefit, because instead of turning the T cell off, I turn it on and turn it against these cancer cells to trigger that cancer cell death. So let's talk about that. So anti-PD-1 and PD-L1 inhibitors, let's kind of break these down into their categories. So let's first talk about the PD-1 inhibitors. And then after that, we'll go over some of the names for the PD-L1 inhibitors. All right, so let's say here we have a T cell. On the T cell, what does it express? It expresses something called a T cell receptor. That is what interacts with the antigen that's presented on this kind of cancer cell. The antigen is usually expressed on a MHC molecule, and the MHC molecule interacts with this cluster differentiation protein on the T cell. The other component on the cancer cells expresses something called PD-L1. And then on the T cell, it expresses something called PD-1. We already talked that whenever these interact, what does it do? It turns the T cell off. I want to turn the T cell on. Don't get nasty, but that's what I want to do. So I'm going to give a PD-1 inhibitor or a PD-L1 inhibitor. With PD-1 inhibitors, you can, again, remember it ends in MAB. The MAB's portion of this is, you can think about pembrolizumab or nivolumab. For PD-L1 inhibitors, you can think about atezolizumab or durvalumab. Now, again, I don't want you to go too crazy. You don't have to memorize all of these. I just want you to remember that these are the ones that end in MAB, and that these can be PD-1 or PD-L1 inhibitors. Now, if that is the case, if I give these drugs, the PD-1 inhibitors, when I give these monoclonal antibodies, will bind to the PD-1, right? They'll bind to the PD-1 or they'll bind to the PD-L1. If they do that, essentially what is happening is if you bind to the PD-L1 with a PD-L1 inhibitor, it won't be able to interact with the PD-1. If you bind to the PD-1 and you block it, you won't be able to interact that with the PD-L1. Essentially, you are inhibiting this connection. This is the very connection, essentially, son of a gun, that does what? Turns the T cell off. If I block this connection, what will I do? Turn the T cell on. So that's what I want to do. I want to give PD-1 inhibitors or PD-L1 inhibitors. And this is essentially going to do what? They're going to be monoclonal antibodies that bind PD-1 or bind PD-L1 and inhibit this interaction. Essentially, what will that do? That'll take and turn the T cell on. If I turn this T cell on, that is going to trigger the T cell to be able to cause death of these cancer cells. So it'll then trigger cancer cell death. And that's what we want. We want these T cells to fight the battle. But we just need to help it by giving these immune checkpoint inhibitors. Now, in the same concept, but we're going to be a little bit different in the mechanism, you have anti-CTLA-4 antibodies. Now these, usually the one that I like to remember is ipilimumab. And again, just remember the MAB portion. With this, it's a monoclonal antibody, but then you got to talk about, okay, how's it really working? Well, here we're going to have this blue cell. This is a antigen presenting cell. This is a T cell. Again, think about the connections that we have going on here. What is this red one here? This is a MHC complex, probably type 2. There's an antigen. What's it binding with? The T cell receptor, right? Then cluster differentiation protein from the T cell interacts with the MHC. Over here in green on this APC, you have what's called B7. This black protein is called CD28, and this blue one is called CTLA4. Whenever CTLA4 is bound to B7, it inhibits the T cell. Whenever CD28 is bound to B7, it leads to co-stimulation and activates the T cell. I want these to interact. So what do I gotta do? I gotta bind up the CTLA4 so he can't interact with the B7. So that's what I'm gonna do. I'm gonna give this drug, the anti-CTLA4 inhibitor, and by doing that, I'm gonna give these monoclonal antibodies. And these monoclonal antibodies are gonna go do what? They're gonna bind all over this thing. And now it's not allowed to interact with B7. So who now is open to come to the party? CD28. And because of that, this allows that co-stimulation signal that we've talked about, son of a gun, all the way back to immunology when we talked about co-stimulatory signals. This thing gets activated. So then what happens? We activate the T cell. We turn on the T cell. If we turn on the T cell, what will it do? It'll trigger this cancer cell death that we want. So when we talk about ways that we can target different cancer cells, think about it, man. We can target the cell cycle by targeting maybe the S phase. We want to target them as we're trying to incorporate making nucleotides or taking the nucleotides and incorporating into the DNA. We want to block DNA replication by doing it at the supercoil, by also potentially maybe intercalating, by forming covalent bonds, by generating free radicals. We want to then inhibit, let's say, we make proteins that are essential to separate the DNA chromosomes, or the chromosomes, which is the genetic material between the two cells. We can use microtubule inhibitors. If we want to be able to come up with ways to inhibit the downstream cascades that push the cells into the cell cycle via the CDK pathway, we can use tyrosine kinase inhibitors. We can use JAK inhibitors. We can do HER2 pathway inhibitors. We could do what else? You could do MAP kinase inhibitors, CDK4/6 inhibitors. If I want to use hormonal therapy, I could use things like GnRH agonists, GnRH antagonists, SERMs, aromatase inhibitors, anti-androgens. The list goes on. And if I want to activate the immune system and have it fight my battles for me, I can give things like rituximab, or I can give things like CTLA4, anti-PD1, anti-PDL1 inhibitors. And if I want to starve the cancer of its blood supply, I can give things like bevacizumab. You see how these are so many different mechanisms that we can use to target. And that doesn't even, believe it or not, we didn't talk about all the anti-neoplastic agents that you could talk about. I just talked about the ones that you probably could see on the exam and the ones that really make sense with these mechanisms. What I want to do is, because believe me, I understand the overwhelming feeling that you probably have right now with all of these different agents and their mechanisms. Let's go through this step by step. Let's do a quick review of all of these, looking at it holistically as a quick summary. And then from there, we'll move on to the next step, which is talking about the indications. When do you give this drug? Or more likely, because patients who have cancer, we're treating them with combination chemotherapy, we'll talk about those drugs, because you'll learn later when we talk about the basics of chemotherapy, when you give a drug, you can have, let's say you have a tumor, 1 gram tumor in your lung. When you give that chemotherapy, your goal is to have that log kill hypothesis, to kill a constant fraction of cells with each dose until you shrink the cells down to nothing. That's curative chemotherapy. Sometimes you can do that. You can treat a cancer with just chemo. Sometimes you can't do it completely and you need a little help. So then you got to cut the tumor down, debulk it. That's surgery. And then afterwards, you have some residual tumor cells left. You give chemo until they reach curative points or remission, right? So that's curative chemotherapy. Sometimes you have a patient who has a mass that's so big, it's spread to other areas of the body. Your goal is not going to be getting rid of it, it's going to be reducing the size of it and at least kind of slowing the progression. So if you can decrease the size, cool, but if you can at least prevent its progression from getting bigger or give them a little bit of a better quality of life in the meantime to reduce symptoms from the size of it, that's what we call palliative chemotherapy. And so we'll talk about all that when we get into the combinations and different therapies. And then once we do that, let's come back and let's talk about some of the adverse adverse effects, because when you give these drugs, you have to consider what you need to monitor for. Let's say that we kind of go through our cell cycle. Obviously, we have some cells when they go through the cell cycle, they don't kind of just go back in and go through the replication process. Some of them kind of just live in that G0 phase where they just kind of, they rest, right? They're in that quiescent phase. Some of them though, they actually run right into the next part where we're actually going to start getting ready for that genetic material to get ready to be replicated. We're going to start kind of beefing up some of our organelles. We're going to start getting increasing protein synthesis that we need during that G1 phase. Then what happens is we're going to get these chromosomes ready and we're going to go ahead and replicate them. So we're going to undergo that DNA synthesis process that occurs in the S phase. Now, after we replicate that, we got to start getting a lot of proteins and enzymes ready that are going to help to allow for the separation of this cell from one overall cell to two daughter cells. And so that's when we kind of start getting the actual cell ready to do that. And that's in that G2 phase. What happens is after this cell kind of gets ready, we're then going to start going through the steps of separating the cell. So we're going to go through prophase, we're going to go through metaphase, anaphase, telophase, and then we're going to pop out another cell. So you see how from this, we start with one cell and we kind of continue throughout this process and we can pop off two new daughter cells. That's the goal of the cell cycle. So what we know about neoplasia, what we know about cancer is that these cells continue to really drive themselves through the cell cycle very aggressively. If there's ways that we can come up with drugs to target these cells at maybe specific phases or at any phase of the cell cycle, that is the goal of a lot of these antineoplastic agents. Let's kind of review them. So real quick, before we do that, just you have to remember there is another point. So there is checkpoints along the way. One that's really important is the G1S phase checkpoint. That's really, I'd say, highly dependent upon something called CDK, cyclin-dependent kinases. And so it is important to remember those. Those work really importantly in that G1S phase checkpoint. And we'll talk about a lot of different drugs that work at that point as well. Let's dig into it. First thing I need you guys to remember is that we have purines. These are a specific type of nucleotides. We have pyrimidines. These are nitrogenous kind of bases. And these are going to be helpful for making nucleotides. So we have purines and pyrimidines. What's going to happen is we use a lot of different enzymes in these processes that we talked about to make ribonucleotides, which are basically, as you see here, a phosphate group, a ribose sugar, and then the appropriate purine or pyrimidine. All right. Now, the enzymes needed, there is a lot of them. Just a couple of them that we talked about is like PRPP synthetase. Another one is called adenosine deaminase. There's another one called dihydrofolate reductase. And then there's what's called thymidylate synthase. These are some of those enzymes that we talked about in individual portions that are critical for taking purines and pyrimidines and helping to make ribonucleotides. Now, ribonucleotides, they're very important. They're a base structure that all we want to do is take this hydroxyl group and get rid of it and turn it into a hydrogen. And that converts this into a deoxyribonucleotide. This process is dependent upon specific enzymes. And that's specifically what we call ribonucleotide reductase. The other thing is that we want to take each one of these deoxyribonucleotides in the form of adenosine, guanine, thymine, and cytosine, and really incorporate them piece by piece into the DNA. And the person who facilitates that is the DNA polymerase enzyme. And then the other thing is that we don't want the DNA to over twist and supercoil, so we use topoisomerases, type 1 and type 2, to help to maintain that DNA structure. And then what we know that DNA can do for us is that we can transcribe it to make RNA, and then RNA can then be utilized in the process of translation to make proteins. And one of those proteins that are super critical to the cell undergoing its whole proliferation process is microtubules, as a specific example here. So now, I kind of want to take this generic diagram and really apply how a lot of our antineoplastics works in more of a simple way if you want a quick reminder. Let's go back. Let's talk about the S phase inhibitors. That was a lot of your antimetabolites. And there was a bunch of them. If you guys remember, 6-mercaptopurine and azathioprine, they were really powerful kind of drugs that can help to create these intermediates, right? The thio kind of purine kind of like analog And what's important about that is that there was really critical to inhibiting the PRPP synthetase and the adenosine deaminase, right? So it was really crucial with the IMP process, that pathway. And so we're inhibiting that. And what happens is that's going to inhibit the purines being converted into ribonucleotides, being converted into deoxyribonucleotides. You get less DNA that's being made. And if you can't make DNA, you can't replicate the cell. That's a really cool process there. Antimetabolites like methotrexate, 5-FU, which is your 5-fluorouracil, these were critical to playing a role with the dihydrofolate reductase. That's specific to the methotrexate. And the 5-FU was specific to inhibit the thymidylate synthase. And these were critical, particularly if you know methotrexate, it's important within the folate pathway. Now, folate pathway does help to make purines, and it also is really important in making pyrimidines like thymidylate. And so that's really, really important to remember that. And so when we inhibit these enzymes, we then essentially inhibit the formation of pyrimidines. And if you can't make pyrimidines, you can't make ribonucleotides. And you can't make deoxyribonucleotides, you can't make DNA, and you can't replicate the cell. Other drugs that we talked about is like hydroxyurea and gemcitabine. These are drugs, so hydroxyurea is a pure ribonucleotide reductase. Gemcitabine has multiple types of functions. It's just one of its additional functions is it can inhibit ribonucleotide reductase. What did that enzyme do again? It took a hydroxyl group from a ribonucleotide and then converted it into a hydrogen group. If we inhibit that enzyme, we can't do that. We can't make deoxyribonucleotides and we can't incorporate them into the DNA. If we can't make DNA, we can't make new cells, my boy. Now, another antimetabolite groups is going to be cytarabine, and these ones are cool because we can kind of group these together. Gemcitabine, which we already talked about, is right here, which is an RNR inhibitor. Another one is cladribine, and then lastly is fludarabine. These are what we can kind of refer to as nucleoside analogs, if you will, in the sense that what they can do is they can kind of be incorporated as like a fake nucleotide, all right? They come in as nucleoside analogs. And then what happens is once you try to add them into the DNA, they go through like a phosphorylation process. Once you try to incorporate them into the DNA polymerase can't click. So it takes the drug, the cytarabine, the gemcitabine, the cladribine, the fludarabine. They're going to act like a nucleotide. They're going to get clicked into the nucleotide prior to make a DNA strand. And then whenever the DNA polymerase goes to grab the next kind of nucleotide, it can't click it together. And that terminates the DNA replication process, which is, I think, really cool. Also, some of these, especially gemcitabine and even cytarabine, can inhibit, and a lot of them can inhibit DNA polymerase itself. So they can inhibit it directly or indirectly by inhibiting DNA polymerase to add another nucleotide onto itself. So that's a really cool concept to think about. Now you see here that antimetabolites, again, a lot of them, and we can think about them in this particular way, or we can think about them in this diagram that we talked about. It's very simplistic, but these really help to inhibit the cell within the S phase when DNA replication is crucial. And so that's going to help to inhibit a ton of cell replication, which is great in a lot of cancers, especially kind of like any cancer that is involving the bone marrow. And we'll go through a lot of indications and when these kind of drugs can be utilized. But these are really, really powerful. And so because of that, they can also affect any other cells that are highly replicating. So like epithelial cells, your bone marrow cells. They can affect any kind of mucosal tissue. And so that's why you get a lot of side effects, unfortunately, with these drugs. But another way to group them, as we talked about, is that methotrexate is a folate antagonist. 6-mercaptopurine, azathioprine, cladribine, and fludarabine specifically are inhibiting the purine pathway. And then your pyrimidine antagonists are going to be your 5-fluorouracil, cytarabine, and gemcitabine. And then your ribonucleotide reductase inhibitors is primarily hydroxyurea, with the addition that, as we have asterisks here, gemcitabine can also be a ribonucleotide reductase inhibitor. All right, that's your antimetabolites. That's the beefy group. Let's now move into another group. When we think about these topoisomerase inhibitors, they're actually pretty cool how they kind of work. So again, you have etoposide, teniposide, irinotecan, and topotecan. And again, we talked about this a lot. All I want you to do is remember how are they kind of like working in this process. They're going to inhibit topoisomerase enzymes. Topoisomerases are critical for the DNA supercoils. They're kind of preventing the DNA supercoils. That's how the enzymes generally work. If we inhibit those, we allow for these supercoils to either get worse, or we incorporate a lot of DNA fragmentation into the process, and essentially what happens is whether you fragment the DNA or you inhibit the actual kind of you basically continue to induce supercoils, if you will, that inhibits the DNA replication from occurring at the replication fork process. So either way, you're inhibiting the DNA formation. Another thing I want you to remember is anti-tumor antibiotics also work at this level. So this is going to be like doxorubicin, daunorubicin, dactinomycin, bleomycin. These are going to inhibit the DNA from actually being able to replicate or at least form. The mechanisms, if we're really being particular, again, doxorubicin, daunorubicin, dactinomycin, they really have that three-prong mechanism. They can act as an increasing reactive oxygen species. They can kind of intercalate in kind of the DNA and form like weak bonds between the nucleotides that inhibits the replication fork and the DNA polymerase from moving down it. And on top of that, they also have topoisomerase type 2 activity. So they can even kind of like, again, cause the topoisomerase to cut but not to be able to religate. And that's where you get that fragmentation. You also, again, continue allowing the supercoils to occur. All of these things inhibit the DNA from being properly replicated. Another thing is alkylating agents. And so this is your cyclophosphamide, ifosfamide, melphalan, temozolomide, cisplatin, carboplatin, oxaliplatin. All of these are primarily working to kind of like get into the DNA and to cross-link. They really form very strong kind of like cross-linking between the actual nucleotides, whether it's interstrand or intrastrand. And the problem with that is, then again, it's the same thing. You can't allow for the DNA polymerase to move down the DNA and allow for it to continue to replicate and allow for that process to occur. So that's one thing. You don't allow for the replication forks to be opened up. All of these things, they inhibit DNA from being formed. So all of them kind of work at that step as a simple way of remembering it. But then to kind of take it to the next level, topoisomerase type 1 inhibitors exist. That is technically the irinotecan and topotecan, and they really only work best in the S phase. Topoisomerase type 2 inhibitors, as you can see color coordinated here, this is etoposide and teniposide. They can also work in the S phase, but they really are more prominent in the G2 phase. And then lastly is bleomycin. It's one of the only other kind of antitumor antibiotics that are listed that really has a cell cycle specific phase that it works in, and that works in the G2 phase. As you can see here in all of these like colors, antitumor antibiotics like doxorubicin, daunorubicin, dactinomycin, cyclophosphamide, ifosfamide, melphalan, temozolomide, all the platinum compounds, these are cell cycle non-specific. So antimetabolites are S-phase specific. Topoisomerase type 1 is S-phase specific. Topoisomerase 2 is G2 phase specific. Bleomycin is G2 phase specific. All of these other agents that we have right here that are not bleomycin in this scenario, that are kind of in this darker gray-black color, these are cell cycle non-specific, meaning they can work at any phase. They can work at G1 phase, S phase, G2 phase, M phase. They could even work in the G0 phase. It doesn't matter. That's an important thing to take away from that. The last one that is a cell cycle specific agent is going to be your microtubule inhibitors. This is going to be vincristine, vinblastine, docetaxel, paclitaxel. And what they do is they inhibit particular proteins in this kind of like overall simplistic diagram. Remember, they inhibit the microtubules. They either inhibit the polymerization reaction or the depolymerization reaction. The whole point of that is that you can't allow for the cells to go through their proper metaphase, anaphase, telophase process. And so if we really want to be particular, microtubule inhibitors can be broken down into two groups: the vinca alkaloids, which is your vincristine, your vinblastine, and then the taxanes, which is your docetaxel and paclitaxel. And the thing to remember about that is that the vincas, those are working to inhibit more of that kind of polymerization, and then the taxanes are working to inhibit more of the depolymerization process. That really gives you a good understanding of the cell cycle specific and cell cycle non-specific agents that can either work in a specific phase or any of these phases. What about agents that maybe work more particularly at like the G1S phase checkpoint? So we're kind of going to be a little bit specific, but also kind of indirect. And we kind of talked about this a lot, where we have something called CDK4 and 6, cyclin-dependent kinase type 4 and type 6. Now, what I want you to remember is there was a lot of different receptors that were on a cell surface, and especially in this particular scenario, a cancer cell. You have things like JAK2 receptors, epidermal growth factor receptors, you have ALK receptors, you have BCR-ABL proteins, so they're not really receptor, they're kind of a non-receptor tyrosine kinase enzyme, and then you have HER2 receptors. You also have estrogen, which is really, really important in some types of tumor cells, and they work on particularly estrogen receptors. These are usually intracellular. They are not usually extracellular, as we've seen with these particular scenarios. And then you also have androgens. Androgens are really important in certain types of tumors as well, especially like prostate cancer. And they also have intracellular receptors that they bind to, like androgen receptors. What you'll notice is that there's a common theme, and that all of these usually end, at least in some way, shape, or form, on cyclin-dependent kinase type 4 and type 6. And that's really, really important, because what I want to do is I want to go through the left half and I want to show you how they lead to cyclin-dependent kinase. Then I'll go through the right half and show you how they lead to cyclin-dependent kinase. So first thing here that I want you to remember is that there's 3 intracellular pathways that are super crucial to then activate cyclin-dependent kinase on this left half of the illustration. Let's kind of go through this. One is called PI3K and AKT. This is an intracellular pathway that is really critical, that helps to be able to activate specific transcription factors that induce the cell to undergo growth. It activates proto-oncogenes, inhibits tumor suppressor genes, and essentially causes the cell to go through its proliferative processes. Another one is called the signal transducer activator of transcription. Whenever you have these receptors or these pathways, they stimulate this transcription factor to do the same thing. Stimulate the proto-oncogenes, inhibit the tumor suppressor genes. And then also you have what's called RAS, RAF, MEK. This is all going to lead to your MAP kinase pathway. And that's, again, going to activate particular transcription factors that stimulate proto-oncogenes and inhibit the tumor suppressor genes and push the cell to go into its replication process, especially in cancer cells if all of these are hyperactivated, right? So let's say that a person is going to be, they receive some type of like epidermal growth factor and it binds to that receptor. It can then work to increase this pathways. ALK, if you have some type of a growth factor that binds to this tyrosine kinase receptor that's usually has an ALK rearrangement, that can also increase this pathway. If you have things that bind to BCR-ABL, usually things like ATP, it activates this enzyme and helps it to then push it into this pathway. Or if there's mutations in that enzyme, which causes it to be constitutively active, that can then push it to kind of activate these pathways. If there's ligands that bind onto the HER2 receptor, that can stimulate these 3 pathways. Now, JAK2 is a little bit more specific, so that's a Janus kinase. Whenever it's activated by specific growth factors, it can really inhibit more of a particular pathway. And then again, whenever you have like ligands binding to the JAK2 proteins, again, this is going to trigger an intracellular pathway that's going to activate, again, that STAT, that signal transducer activator of transcription. Here, what I want you to do is we're going to see kind of a common theme. Estrogen, whenever it's in high amounts, can bind to the estrogen receptor. And when it binds to the estrogen receptor, it's going to activate a specific enzyme. Androgens in high amounts will bind to the androgen receptor, and when high amounts are going to activate this particular enzyme. Whenever you have these growth factors, and in patients who have maybe specific types of mutations in these receptors, they will then increase these 3 pathways. All of this will converge onto one thing. And that's this enzyme complex called CDK4/6, right? So there's cyclin-dependent kinase 4 and cyclin-dependent kinase 6. And these are usually associated with a cyclin enzyme. Once this kind of activates this enzyme is going to do what a kinase does. It's going to phosphorylate proteins. And so what it does is it puts phosphate groups onto the retinoblastoma protein. Retinoblastoma protein, think about it, basically it holds E2F normally. If you add phosphates to it, its mouth opens and E2F goes from a bound form to a free form. Whenever you have free amounts of E2F, this acts essentially like a transcription factor that then goes down, activates those proto-oncogenes, shuts down those tumor suppressor genes, and then does what? Causes the cell to go through excessive replication cycles and grow, and that's how you end up getting cancers. So that's why patients who have specific types of JAK2 mutations, they're predisposed to specific cancers. People who have mutations in these receptors, they're predisposed to specific cancers. HER2, predisposed. And the conditions of where you have high amounts of estrogen, it predisposes you to the cancer growing or getting worse. High amounts of androgens, conditions that either increase your exposure, risk, or progression of that particular cancer. So then what we need to do is kind of like figure out how can we kind of insert these drugs along this process to inhibit these. So let's kind of think about that. What I want to do is I want to, again, tell yourself the cell can go into a G0 phase where it just kind of rests, right? Doesn't really do anything. It's not going to actively kind of replicating, or it can decide to go into the replication cycle and then proceed to the G1 phase. Then it's going to replicate its DNA in the S phase. It's going to get ready to start processing the mitosis in the G2 phase. And then it's going to run through mitosis and make another cell. Again, these agents that we're going to talk about aren't cell cycle specific. They're kind of like non-specific, but they really tend to work at checkpoints in an indirect manner. And that's what I really want to talk about. So one of these could be JAK inhibitors, that's specifically a JAK2 inhibitor. This would be an example like ruxolitinib. And it's really going to inhibit what? Well, it's going to inhibit the JAK2 from being able to send this signal. And if you don't send that signal, what's going to happen is you're not going to have an increase in the stat. You can give tyrosine kinase inhibitors that actually work against that EGFR. This would be like osimertinib. If you shut down that receptor, you don't send those signals. If I use things like an tyrosine kinase inhibitor like alectinib, that's going to shut down that receptor and it's not going to send those signals. If I can use things that inhibit the BCR-ABL enzyme, I can give BCR-ABL tyrosine kinase inhibitors like imatinib. And that's going to, again, shut down that enzyme from sending the signals. If I can give things that are HER2 inhibitors, this would be something like trastuzumab. That's going to shut down the signals going from that receptor. And the beautiful thing about this, if you guys remember, is this is a monoclonal antibody. These are protein kinase inhibitors. This is a monoclonal antibody. Again, you see a pattern here that if we're inhibiting these receptors, it's going to shut down these intracellular pathways. If we shut down these pathways, we then drop the levels of PI3K, AKT. We drop the levels of STAT. We drop the levels of the RAS, the RAF, the MEK. Now, these enzymes have the ability to go and work on the DNA themselves. But in this pathway, if you don't have enough of these, are you going to be able to stimulate CDK4 and 6? No, and that's a problem. One other drug that I want to mention is that this RAS, RAF, and MEK, we can actually give drugs that directly inhibit the RAF. And that's called your BRAF inhibitors. That's vimurafenib. So that can directly inhibit this particular pathway with emphasis on the RAF. Either way, you get the point. If I don't have these guys that are in high amounts and they're actually in decreased amounts, they're not going to be able to stimulate CDK4 and 6. So that CDK4 and 6 level is going to become less active. We're going to inhibit it theoretically. Now, another thing that's really important is that we have drugs that can directly inhibit CDK4 and 6, and that is usually your CDK4/6 inhibitors. This is like your ciclibs, right? So palbociclib. And what happens is they can directly inhibit this enzyme. So these are indirectly in some way inhibiting this enzyme. This one is directly inhibiting this enzyme. Now, the beauty of this is that if you inhibit this enzyme, what happens? You no longer phosphorylate retinoblastoma protein. Can it release the E2F? No. What happens to the free levels of E2F? It drops. What happens to you now stimulating proto-oncogenes? Nope. What happens to you inhibiting tumor suppressor genes? Nope. So now, in this scenario, you're not allowing for the cell to replicate and you're preventing the cell from continuing to evade apoptosis. That's going to shut down that cell growth. And that is a way that we can kind of control the cancer. All right, so now, again, what we kind of see out of this is that if we're theoretically inhibiting this cell in that G1 kind of S phase checkpoint, that's really where it's working. It's not working in the G1 phase. Again, it's working in the G1 S phase checkpoint. Then what we're going to do is we're going to theoretically inhibit this cell from going through the S phase, the G2 phase, the M phase, and then we're going to drop down that cell proliferation the simplistic way, right? So that's what I want you guys to think about here. Now let's move on. Let's now cover the right arm. So we went through the left arm. Let's go through the right arm of this scenario. Again, I want you to remember that androgens, they are capable of doing what? Being converted into estrogen. Through what enzyme? Aromatase. And that's a really important thing to remember. And then we know that estrogen can bind to estrogen receptors and they can act on CDK4 and 6 and they kind of stimulate that pathway. What if in that scenario, I think about ways that I can target specific areas. So I'm going to use something called SERMs. These are your selective estrogen receptor modulators. This is going to be things like tamoxifen and even technically to a degree raloxifene. And what happens is these are going to help to inhibit the interaction between estrogen and the estrogen receptor. So if you inhibit that, you essentially inhibit this ability to do what? Stimulate the CDK4 and 6, right? Another drug that we can give is GnRH agonists. And this is usually going to be things like leuprolide. And then we have GnRH antagonists, which is going to be like degarelix. And what these are going to primarily do is they're going to inhibit, they do it in specific ways. So GnRH agonist is going to lead through a kind of a process where it increases FSH and LH, and that's then going to go and cause an increase in estrogen, but then there's going to be a negative feedback that then shuts down the FSH, shuts down the LH, and eventually drops the estrogen. That's how GnRH agonists would work. GnRH antagonists, they're going to specifically work in what way? They're going to directly inhibit FSH and LH, and that's going to lead to a drop in estrogen. The combination of both of these do what? They drop estrogen. If you drop estrogen, then what? Can it activate a receptor? No, not if you don't have enough of it. Can it then stimulate the CDK4 and 6? No. And so that's a problem. Aromatase inhibitors are another one. This is usually things like anastrozole, letrozole. And what they do is they inhibit the aromatase enzyme. If you inhibit the aromatase enzyme, can you convert androgens into estrogen? No, so you effectively lower the estrogen levels. You don't have enough estrogen to bind to the receptor. Can it activate the CDK4 and 6? No. Now, you get the point that all of these influence a drop in estrogen. That drop in the effective estrogen activity or the ability of estrogen to bind to the receptor leads to an inhibition of the CDK4 and 6. Anti-androgens are another one that we can use. And what they're going to do is they're going to block androgens from binding to the receptor. And that's usually like enzalutamide. And what happens is if you do that, you don't allow for androgen to bind to the receptor. That complex is no longer able to help to activate the CDK4 and 6. And so essentially you're inhibiting CDK4 and 6. If I inhibit CDK4 and 6, what happens again? We know that we don't phosphorylate retinoblastoma. We don't release the E2F. We then continue to inhibit proto-oncogenes. We stimulate the tumor suppressor genes, and we stop the cell from continuously growing. And that is going to stop that cell proliferation. That's going to help us with these particular cancers. Now, again, we're going to go through the indications in a little bit, but I think what's kind of cool about this is that you can kind of think about how these kind of like drugs work. And so I want you to think about this. Usually BRAF inhibitors are great within melanoma. JAK2 inhibitors are really good within patients who have something called polycythemia vera. It also is good in conditions like myelofibrosis. And again, these are usually a myeloproliferative neoplasia. And then what we'll see is that lung cancer, these really respond well. Any kind of EGFR tyrosine kinase inhibitor and ALK tyrosine kinase inhibitor, with their specific mutations present, they will respond well in patients who have specifically non-small cell lung cancer. We know that BCR-ABL tyrosine kinase inhibitors are really good in those patients who have the Philadelphia chromosome, the 922 translocation, because it basically creates a mutation in this BCR-ABL. And so really, really good in CML. And then in some small cases of patients who have AML with the 922 translocation, it can also be utilized. HER2 is really, it can be beneficial in metastatic gastric and esophageal cancer, but what we commonly know it utilized in is it's going to be breast cancer. Now, we also know that breast cancer is really, really important because you can use breast cancer as a way of saying we can use CDK4 and 6 inhibitors. We can use SERMs. We can use aromatase inhibitors. And obviously in these certain scenarios, it depends upon if they're estrogen receptor positive, progesterone receptor positive, if they have metastatic scenarios, if they're premenopausal, postmenopausal, if they have HER2 positive breast cancer or not. There's a lot of factors. And again, we'll talk about that a little bit later. And then for your GnRH agonists and your antagonists and the anti-androgens, these are really going to be beneficial in patients, especially those who have things like prostate cancer. And so that's something that I want you guys to think about in this potential scenario. Another potential thing I want you to think about is we talked on the whiteboard about immune evasion. We talked about angiogenesis. Let's kind of just have a quick little recap of that. What we know is that these cancer cells, again, they're releasing something called VEGF, right? Vascular endothelial growth factor. And what that does is that helps to activate those endothelial cells of nearby blood vessels to start making more, going through the replication and making more endothelial cells, which then kind of sprouts these little branches off of the artery. And that's called angiogenesis. Whenever they bind to that receptor, they promote a formation of new blood vessels. If you have more blood vessels, you theoretically have more blood flow. More blood supply to a cancer just allows for that cancer to continue to keep growing and growing and growing. Now, what if I give a VEGF blocker? That's something like bevacizumab. It's a monoclonal antibody that binds to VEGF, prevents it from binding to the VEGF receptor. If I can't bind to the VEGF receptor, I can't undergo the angiogenesis process and I can't actually increase blood supply. So theoretically, I starve the cancer cells, and that may, over time, promote cancer cell death. Theoretically, right? Now, another concept here is that some patients, especially who have cancerous cells, and I want to be specific and say cancerous lymphocytes. Cancerous lymphocytes, sometimes what can happen is they can express specific types of proteins on their surface, right? Especially CD20. And we see this a lot in patients who have what's called non-Hodgkin's lymphoma. When you express CD20, we can actually use a drug called rituximab, which is a monoclonal antibody that can bind to the CD20. And then it can promote three mechanistic responses. One is it can promote a complement fixation, which can lead to opsonization, or worst case scenario, it can lead to the complement proteins promoting what's called a membrane attack complex. Membrane attack complexes are pore inside of the actual cell that then allows water to rush in and induces lysis of that respective cell. On top of that, it can actually just initially activate apoptotic mechanisms in the cell, may undergo apoptosis. And on top of that, it may then activate specifically natural killer cells to then see these particular antibodies bound to the CD20, and it may induce it to undergo a process where it releases things like granzymes. And when it releases those granzymes, that then triggers the cell death process, and that may cause these cells to die. So that's one way where we can use rituximab to treat a particular cancerous condition, especially in situations where you have lots of cancerous lymphocytes, like in non-Hodgkin's lymphoma. All right, so the next thing I want you guys to think about is when we have these cancer cells, these cancer cells are finicky. They're kind of smart where they can kind of change expression of specific molecules, right, on their surface. And they can also change the expression of certain molecules on even other cells like T cells. And so they're kind of cool. Terrible thing, but we have ways of we figured it out. We're a little bit smarter than the cancer cells kind of, right? So cancer cells, what they can do is, again, they express something called PD-L1. And a lot of the times what happens is that they can really overexpress that in significant amounts. And you'll see why in a second. Again, we already kind of talked about this with a nice little recap. MHC complexes are pretty common, especially more of the type 1 on their surface. They usually present it with some sort of an antigen. And then T cells, we know that they interact with usually these two proteins in specific ways. So PD-L1 interacts with PD-1, and then T cell receptors usually interact with the antigen, and they have a CD protein that interacts with the MHC complex. Whenever you have this interaction between the PD-L1 and the PD-1, when they click together, it kind of turns the T cell off, and it has no desire to want to do anything with this cancer cell. So it's kind of smart in that way that if a cancer cell expresses a lot of the PD-L1, it creates a problem. Another thing that T cells do is they express something called CTLA4. And what happens is that's a normal molecule, but cancer cells, they lead to the overexpression of CTLA4. T cells, again, we know they express T cell receptors, and we know they express something called CD28. What I want you to remember is that CTLA4 and CD28 are kind of like competitors for another protein on a cell that's going to pop in called an antigen-presenting cell. Antigen-presenting cells, they interact, again, we already know, MHC complex, which has a little antigen on it. Usually these are type 2. They interact, again, that antigen with the T cell receptor, and then there's a CD protein, usually that interacts with the MHC complex. But here's the big competitor. This is called B7. B7 has an opportunity to interact with CD28 or CTLA4. If CTLA is present in high amounts, it's going to have a higher affinity, and they're going to interact. When they interact, the T cell is then what? Turned off. But whenever B7 and CD28 interact, the T cell is turned on. So that's what we want to kind of think about here. So again, PD-1, PD-L1, when they interact, the T cell's turned off. When CTLA4 and B7 interact, the T cell's turned off. What I want to do is I want to come up with ways of being able to inhibit this interaction to turn the T cell on. And I want to inhibit this interaction to turn the T cell on. So how am I going to do that? Well, if I give a PD-L1 inhibitor, what it'll do is, this is atezolizumab and durvalumab, these will then come in and they'll kind of bind their monoclonal antibodies, and they'll bind at the PD-L1 and block the PD-1 from being able to kind of click in. So that's one way. Another one is I could just give a PD-1 inhibitor. And this is going to be things like pembrolizumab, nivolumab. They're going to come in, act as a monoclonal antibody, and bind to PD-1 and inhibit PD-L1 from clicking into it. That's going to inhibit that interaction. And that theoretically would then allow the T cell to be turned on, and then it's going to want to kill the cancer cell, right? Another drug that we can give is a CTLA4 inhibitor. If CTLA4 is increased in expression on cancer cells, if I block it with an antibody like ipilimumab, what's going to happen? Well, then it's no longer going to compete with CD28 with B7. So who's going to interact a lot? The CD28 and the B7 will start to interact. And if they interact, at least the B7 ain't interacting with the CTLA4, and that leads to a co-stimulatory signal. That leads to an activation of the T cell. And that T cell going to be ripping them cancer cells up. And that's what I want you to remember, is that the goal is to inhibit that interaction so that we can turn the T cell on. And the same thing with the CTLA4, inhibit that interaction so that we can turn the T cell on. All of this will then allow the T cell to induce that cancer cell death. This is a good recap of all of these mechanisms that we talked about on the whiteboard in great detail. I hope it makes sense, because now what I want to do is I want to move into what are the indications? Why do we even give any of these particular antineoplastics? Well, I want you to kind of think about it in groups. Let's say I talk about hematologic malignancies, so something like non-Hodgkin lymphoma. We use a bunch of different chemotherapy agents sometimes to target these cancer cells. And a common one is the R-CHOP regimen. We talked about that in lymphoma. And that's usually going to be consisting of what? It's rituximab, right? That's going to work against that CD20 molecule. Cyclophosphamide. All right, we talked about that as more of that alkylating agent. Hydroxydonorubicin, which is doxorubicin, that's one of your anti-tumor antibiotics. Oncovin, which is also vincristine, which is one of your microtubule inhibitors. And then prednisone. Prednisone actually naturally has a way of suppressing cancer cells as well. And so it has an immunosuppressant capability, and it's actually going to be used in certain types of scenarios of cancer as well. So you can see how we're using multiple different agents to target one cancer. Hodgkin lymphoma, kind of a similar concept. You can use something called an ABVD regimen, and that is usually going to be adriamycin, bleomycin. So bleomycin is actually one of those agents, which is an anti-tumor antibiotic. Vinblastine, which is a microtubule inhibitor, and dacarbazine, right, which we didn't talk too much about this one. But again, you get the point is that these have the capability of having multiple different agents to target a cancer. It's very common to have combination chemotherapy. Chronic lymphocytic leukemia actually responds pretty well to a BTK inhibitor, which we didn't talk a lot about, but it's called ibrutinib. But it kind of works similar in that it actually is going to have the capability of inhibiting a very specific receptor, the Bruton tyrosine kinase, that's actually present on lymphocytes in these chronic lymphocytic leukemia condition. All right. Chronic myeloid leukemia, we kind of talked about this one as a high yield one. Do you guys remember? It was the BCR-ABL inhibitors. All right, so specifically the imatinib is a really good one. Acute myeloid leukemia, we actually can use combination. Yes, if they have the 922 translocation, we can use this as well. But the big thing to remember is that we like to use cytarabine and daunorubicin. You're like, okay, how do I remember that? Well, a music lover's CD is like maybe a helpful mnemonic for you. So again, AML with a music lovers and CD, cytarabine and daunorubicin. Acute lymphoblastic leukemia is another one that we want to talk about. And this one actually can have combination chemotherapy as well. And usually that's vincristine, prednisone, asparaginase, and daunorubicin. How do I remember this one? Oh, all very powerful agents destroy. So again, you can remember that in ALL, we're going to use vincristine, we're going to use prednisone, we're going to use asparaginase, and we're going to consider daunorubicin in certain types of scenarios. Intrathecal methotrexate is utilized in acute lymphoblastic leukemia prophylactically to prevent the meningeal leukemia because it's a very high risk and it's also has potentially for high mortality. And so we like to give intrathecal methotrexate as a prophylactic measure to prevent meningeal leukemia. All right. So again, ALL in the meninges, you can remember intrathecal methotrexate. And again, it's a really important one out of all of these to remember. Again, I think it's important to remember these drugs, but it's more kind of giving you a point. The real important to remember for ALL is that we have to give intrathecal methotrexate as a prophylaxis to reduce the risk of meningeal leukemia. Multiple myeloma. This is one that we didn't talk about a ton. Again, it's one of those plasma cell disorders where you're producing tons and tons of plasma cells that lead to a lot of immunoglobulins being produced. But what we like to do with this one is we like to give on something called bortezomib, lenalidomide, and dexamethasone. We didn't talk about this a lot, but bortezomib is actually kind of one of those proteasome inhibitors, and lenalidomide is an immunosuppressant along with dexamethasone. And again, you can remember, dogs love bones. So dexamethasone is for the D, lenalidomide for the love, and then the bortezomib for the bones. MPNs is usually going to be polycythemia vera, essential thrombocythemia. And again, what's really important to remember about these is that these are going to respond well. They can do well with a JAK2 inhibitor, right? They definitely can. But we actually see a better response initially, and it's usually the better way to go about this is with something called cytoreduction. We prefer to give something called hydroxyurea. Do you guys remember hydroxyurea? That was one of those ribonucleotide reductases. It was one of those anti-metabolites. So that's another one. But you also can give, don't forget, you can also give ruxolitinib, which is a JAK2 inhibitor. All right. Now, another kind of group of malignancies is things like genital urinary ones, like testicular cancer. Testicular cancer actually responds well to a combination chemotherapy like the BEP regimen, which is usually going to be bleomycin, etoposide, and a platinum agent. So again, you can see here is an anti-tumor antibiotic. Here is a topoisomerase inhibitor, right? And this is usually going to be a type 2. And then you have a platinum agent. This is an alkylating agent. So again, you're using combination chemotherapy here. Prostate cancer. This one is really targeting that androgen pathway. And so we like to give things that are going to target that. And so we use things like GnRH agonists, which is your leuprolide. That's going to lead to, again, a reduction in particular processes like production of FSH and LH. And again, the importance with that is we didn't talk about this, but LH is crucial for, in the male, testosterone production, which can be converted into DHT, and DHT can actually lead to the enlargement of a particular tumor. It's essentially an androgen, and so that's also crucial. So giving GnRH agonists like leuprolide can inhibit that process, and then giving anti-androgen receptors in worst case scenarios like enzalutamide, that may be able to help these patients. Obviously, the goal is a radical prostatectomy, but this is in combination. Renal cell carcinoma is one of those that a lot of the times it's usually performing some type of surgery. So usually this could be potentially, depending upon the size of it, you may be able to do a partial or maybe a radical nephrectomy. But in metastatic cases, we oftentimes have to reach for specific immunotherapies. And that's where we actually like to dip into a combination of two immunotherapies, usually anti-PD-1 inhibitors and an anti-CTLA-4 inhibitor. All right. Bladder cancer is actually interesting because, again, the goal here is depending upon if it's involving the muscle or if it's not involving the muscle. So usually intravesicular mitomycin C, which is kind of one of those anti-tumor antibiotics, can be used. You can actually indwell the bladder with this particular antineoplastic agent, and it may be able to work against those cancers that haven't invaded the muscle of the bladder. But once it's kind of invaded the muscle of the bladder, you may have to reach for more of the systemic kind of chemotherapy to really prevent the excessive metastasis. And that's when you get onto gemcitabine, which is an antimetabolite, and cisplatin, which is an alkylating agent. All right. Again, you can remember this with the term great combo for the muscle invasive bladder cancer. Again, gemcitabine and cisplatin. All right. What about those scenarios of gynecological malignancy? So things like breast cancer. So in this scenario, we like to really consider SERMs as a beneficial drug. So this is usually going to be those that are working against those who are estrogen receptor positive and progesterone receptor positive, and they're premenopausal. If they're postmenopausal, we prefer the aromatase inhibitors. If they have to add on CDK4 and 6 inhibitors, it's usually in combination with one of these two. So if a patient is, for example, stage 4 breast cancer and they have ER/PR positive, we could do a SERM if they're premenopausal plus a CDK4/6 inhibitor, or if they're postmenopausal, aromatase inhibitor plus a CDK4/6 inhibitor. All right. If they have triple negative breast cancer, in other words, they don't test for ER, they don't test for PR, and they don't test for HER2. That means that this is a patient who has triple negative breast cancer, and they respond well to doxorubicin, which is what? An anti-tumor antibiotic, and paclitaxel, which is a microtubule inhibitor. You can remember no receptors. Okay, triple negative breast cancer because it has no positive receptors. Do Pac-Attack. Right? So doxorubicin and paclitaxel. Anti-PD-1 inhibitors are really kind of one of these niche ones that we can add on. Again, this is pembrolizumab, but usually we have to look at what's called the PD-L1 expression. So this is getting into more particular pathology and immunohistochemistry testing to see if they have a positive PD-L1 expression that's usually greater than 50%. And if they do, they will respond well to inhibiting that PD-L1, PD-1 interaction with pembrolizumab. All right. Ovarian cancer. Ovarian cancer also is one of those that responds, especially since it's a solid organ tumor, usually responds well with, in these scenarios, some type of oophorectomy. Sometimes it may be involving, in certain scenarios, you may have to remove the ovary and parts of the fallopian tubes. In some worst case scenarios, you may even have to remove the uterus. And so sometimes a lot of we see it a lot. Ovarian cancer is a total abdominal hysterectomy with a bilateral salpingo-oophorectomy. And that's usually the standard, but sometimes if it is really severe and it's already metastatic, which is what happens a lot of the times, unfortunately, with ovarian cancer, it's usually doing the best thing you can to do the total abdominal hysterectomy, the bilateral salpingo-oophorectomy, debulking, and then adding on chemotherapy like carboplatin and paclitaxel. And again, you can see this is a platinum agent, and this right here is what? That is going to be a microtubule inhibitor. All right. The next thing is indications of chemotherapy agents for small cell lung cancer, any kind of respiratory malignancy. So small cell lung cancer is very aggressive. Usually by the time it's diagnosed, it's extremely extensive. The goal here is to remember by that time, you're treating them with chemotherapy. They're not going to respond well to any kind of surgical procedures like a lobectomy, pneumonectomy, radiation therapy. At this point, you're probably more likely doing chemo and then maybe doing cranial irradiation therapy sometimes, or prophylactically at least. And so the agents that you'll probably most likely use is cisplatin, which is a platinum agent, and etoposide, which is a topoisomerase inhibitor, usually a type 2. All right, you can remember this by the term elementary cells. So etoposide and cisplatin is going to be great for small cell lung cancer. For non-small cell lung cancer, these are really kind of, oftentimes, for example, in these, chemotherapy is usually going to be the backbone, in combination with determining if you can do things like a lobectomy, if you can do things like a pneumonectomy, if it's maybe areas that you can do like a stereotactic ablation. A lot of the times that's the goal, is to do some type of surgical procedure. But as it starts to get more advanced, then you need to add on things like radiation therapy and sometimes chemotherapy. And usually the base or core of this is cisplatin, pemetrexed, and gemcitabine. Again, platinum agent. And again, here, this is an antimetabolite. We didn't talk about pemetrexed, but again, these are usually your core. And then what I think is important is, as that core, sometimes if the patient has specific mutations that are present, we may add on other things like I talked about. The ALK rearrangement kind of mutation, we can add on things like, for example, for that one, alectinib. If they have the EGFR mutation, we can do things like osimertinib. If they have PD-L1 expression that's greater than or equal to 50%, we can do like an anti-PD-1 inhibitor, like a pembrolizumab. And so those are other things that we add on. But again, I want you to remember the non-small cell lung cancer by cough, puff, or GASP, so cisplatin, pemetrexed, or gemcitabine. So we can use that in non-small cell lung cancer. Again, this is just paying homage that you can add EGFR inhibitors if they have the mutation. So if they have the mutation, osimertinib. If they have this mutation, alectinib. And if they have some type of PD-L1 expression that's greater than or equal to 50%, you can do an anti-PD-1 inhibitor. So that's the whole thing of kind of depending upon the underlying clinical scenario, you're usually using multiple modalities. It's not just chemo by itself a lot of the times. It's often in combination with surgical intervention, radiation therapy that may be used adjuvantly, sometimes neoadjuvantly, and then chemotherapy agents, they can be used adjuvantly or neoadjuvantly, which again, meaning that either neo, it's before kind of a procedure, or adjuvantly after a particular procedure. And so that's something to remember here. Now, what about neurological malignancies? Again, the big one is a GBM. A lot of the particular neurologic malignancies, we don't use chemotherapeutic agents. It's most often, it's surgical intervention is the best way, or treating the associated complications of that tumor. With GBM, it's one of those, again, you do the best that you can to attack this surgically, but oftentimes there isn't much that you can do because of how extensive it is and because of the aggressive nature. But sometimes we will attack this with a combination of neurosurgical intervention and temozolomide, which is a type of alkylating agent. All right. For skin malignancies like melanoma, the best practice is a wide, local excision. So doing a surgical excision, getting really good margins is the best way to go. But on top of that, because melanoma is extremely aggressive and metastatic, you may need to then say, okay, because there is this metastatic melanoma, I need to use chemotherapy agents, because again, the surgical excision will take care of that problem. But at the point of where they have systemic metastasis, you need a chemotherapeutic agent. And usually BRAF/MEK inhibitors are going to be beneficial, but I would say these are more beneficial if you have a specific mutation, because as we talked in skin cancer, we know that patients have specific BRAF mutations that puts them at high risk. And if we can give them vemurafenib, it'll attack those people who have these what's called V600E mutations. But oftentimes, more than not, anti-PD-1 inhibitors would probably be the way to go for any metastatic melanoma. And then you can consider something like a BRAF or MEK inhibitor as an alternative if they have the V600E mutation. What about bone and soft tissue malignancies? We didn't talk about these in oncology. We'll talk about this more in pediatrics, but pediatric osteosarcomas, again, we're going to potentially use these with certain types of therapy, but I think a lot of the times we'll kind of use chemotherapy like methotrexate, which is an antimetabolite, doxorubicin, which is again, what type of agent? This is usually an antitumor antibiotic, and then cisplatin, which is an alkylating agent. And then Ewing sarcoma, this one can be used with a cycle of vincristine, doxorubicin, cyclophosphamide, and then you follow it up with a cycle of ifosfamide and etoposide. So multiple different agents. All right. With gastrointestinal malignancies, we think about colorectal cancer. So what are some chemotherapy agents we use here? FOLFOX is a common one. Again, colorectal cancer, sometimes we'll be doing things like hemicolectomies, partial colectomies, total colectomies. You may need to do particularly like some radiation therapy. A lot of the times you may need to do chemo. In certain situations, you can even use immunotherapy. But in this scenario, I think one of the common chemotherapeutic regimens that are involved is the FOLFOX regimen. So FOLFOX is leucovorin, which is a folinic acid, 5-FU, which is the fluorouracil, and then OX is oxaliplatin. So leucovorin is basically going to enhance the activity of 5-fluorouracil, and oxaliplatin is going to be one of those alkylating agents, is the platinum compounds. Another option is FOLFIRI. So FOLFIRI is again leucovorin, which is the folinic acid, 5-fluorouracil, which again, leucovorin is just going to help to enhance the activity of 5-fluorouracil, and then irinotecan, which is a type 1 topoisomerase inhibitor. Another thing that you can give, again, depending upon if the patient has specific mutations, is the bevacizumab, which is the VEGF kind of inhibitor. Remember, it's the monoclonal antibody, and so it's going to reduce that angiogenic process and starve the actual cancer cell. That's usually if they don't have a specific type of KRAS or BRAF mutation, they're more of that wild type. Cetuximab is going to be one that you actually give, which is a monoclonal antibody that, again, actually helps to work specifically at that receptor, which is one of those EGFR receptors that then helps to stimulate the pathway of developing the RAS and the RAF pathway. And so if that mutation is present, giving something like cetuximab might actually be able to help. And so that's something that you would consider. Anti-PD-1 inhibitors is really if they have something more like Lynch syndrome. So if they have more of that MMR or that microsatellite instability that's present, an anti-PD-1 inhibitor like pembrolizumab may be beneficial. But I really want you to focus on that full sulfonylurea and then to consider these dependent upon those clinical scenarios. All right, then pancreatic cancer, again, we're going to use something called FOLFIRINOX. And again, it's not too bad. Again, we already know that the FOL is leucovorin, which enhances the activity of 5-FU. And then we know that we're going to have irinotecan, which is a topoisomerase type 1 inhibitor. And then we're going to have oxaliplatin, which is an alkylating, again, platinum agent. And that's going to be really good for pancreatic cancer. Again, pancreatic cancer, a lot of the times, we may have to involve a surgical intervention as well, depending upon if it's usually more near the head, which is usually what often happens. You need to do something like a Whipple procedure. And if it's usually near the tail, you may have to just do a tail end resection and sometimes remove the spleen as well if it's kind of close in that area. But again, it's usually dependent upon this, it's combination with the chemotherapy. And we already know that this cancer can be very, very aggressive. But I hope that with that, this makes sense. So now what I want to do is I want to step over into the realm of, again, I'm giving all of these different agents to individuals, but I want to make sure that I'm not causing any kind of toxicity. I'm not causing any kind of like terrible adverse effects, which is going to happen no matter what. But how do I know which agents come with particular adverse effects? And then how do I monitor it? And is there any way in certain types of toxicities, how I address them? How do I reverse them? How do I kind of consider these things ahead of time? Let's actually do that now. All right, my friends, we're almost done. At this point, we've decided we have a patient who's suffering from an unfortunate cancer. We start them on chemotherapy. Maybe it's a combination chemotherapy. And one of the things that we want to be able to do is watch out for any adverse effects. Sometimes you can monitor for these things, and it's really ideal to know that, because that could be a question on your exam is, hey, you put a patient on this particular medication, what should you be monitoring them with? And that could be a way of kind of monitoring for the complication that they could have, right? So we're going to use this Chemo Man, which is a common one that you see in all the different textbooks, the First Aid, AMBOSS, UWorld, it's all over the place and it's well known. And so we're just going to kind of expand on it just a tad. All right, so let's say, for example, we take Chemo Man here and we talk about the ears. Well, we're gonna do this in the shape of what looks like a C, right? And so there's different drugs like cisplatin. So cisplatin is definitely one. And then the other one, it's kind of its cousin, is carboplatin. But I'd say cisplatin is probably going to be the more powerful one here. It can really damage the inner ear. And so it can lead to ototoxicity. So you could develop things like sensory neural deafness. And so that's one thing to definitely consider is ototoxicity. Now, how would you monitor that, right? I mean, is there a way? Absolutely, hearing testing, right? So you can definitely do sensory neural testing. And if you have any confusion of like, hey, I don't know if it's conductive deafness or sensory neural deafness, you can do the Weber-Renay test. From there, determine, hey, they need sensory neural testing to see, do they have any sensory neural deafness, right? So that's one thing to consider there. The next thing that I would say is, let's move from here and let's move down. And let's go and draw this. We're gonna draw the lungs. And they're gonna look like a B. And this is called bleomycin. So this is called bleomycin. Now, bleomycin can lead to something called pulmonary fibrosis. So this can lead to something called pulmonary fibrosis. So pulmonary fibrosis is basically it can generate what's called a restrictive lung disease. And so if you have fibrosis of the lungs, you restrict the expansion of the lungs. And this can lead to some problems with that, right? Now, what would be something that I would want to monitor for a restrictive lung disease? How could I potentially monitor that? I'd say PFTs would be a pretty good start, right? So that's something to definitely consider, is maybe monitor the patient's PFTs. Keep an eye, make sure that they're not kind of moving into that kind of restrictive lung disease type of pattern. So PFTs, DLCO, things of that nature could be potentially beneficial to be checking. All right, so just keep an eye on that. What's the next one? All right, let's move on to the next one. Let's draw something that looks like a heart, but we're gonna do that in the shape of a D, and then we're gonna draw a T in there. All right, so this is gonna be doxorubicin, so doxorubicin. And another one is trastuzumab. And again, I think it's always good to remember, these are alkylating agents, antitumor antibiotics, antitumor antibiotic, monoclonal antibody. What will this do? This is going to act on the heart, boo-boo. And so because of that, this can lead to cardiotoxicity. If it leads to cardiotoxicity, what kind of cardiotoxicity are we talking about? It's usually dilated cardiomyopathy. So that's really the way that you'd want to consider it, is it can lead to something like dilated cardiomyopathy, which you'd have to get an echo. You'd have to be checking some type of like echo process to think about this one. So again, cardio, let's actually fix this one right here. This is cardio. toxicity. All right, so you'd want to be checking a echocardiogram, because echocardiograms are generally beneficial in the sense that we can say, hey, what's the left ventricle doing? Do they have any evidence of like a biventricular systolic like enlargement? Is there a weak or very reduced ejection fraction? And so that's something to definitely be looking for, is looking for biventricular enlargement, looking for a decreased ejection fraction, evidence of any kind of systolic heart failure. All right, the next thing is, so we have this part here. Let's move into the kidneys. And the kidneys, we're gonna do this green. They look like kind of C-shaped. And it's the same group up here. So this could be cisplatin. All right, so this could be like your cisplatin. And cisplatin can lead to something called nephrotoxicity. So nephrotoxicity. And with nephrotoxicity, we worry about kind of a rising creatinine level, right? Or maybe potentially an acute kidney injury. And so what we'd want to be doing for this one is monitoring the patient's renal function tests. I'd definitely be checking things like their renal function tests, whether that be like a BNP, a CNP, or a specialized renal function panel. Either way, check that renal function. All right? You want to make sure that they're not having any rising creatinine or decrease in their GFR, particularly the more the bump in the creatinine because of the risk of AKIs. All right? The next one, let's do this one in orange, given the bladder, and we'll draw this it's a C, and then we'll draw like a Y, kind of like into the urethra portion there. That's going to be cyclophosphamide. So cyclophosphamide. This one is pretty nasty to the bladder. It can lead to something called hemorrhagic cystitis. So this can lead to hemorrhagic cystitis. And in some cases, it can increase the risk of bladder cancer. All right? So hemorrhagic cystitis is definitely something to think about here. With cyclophosphamide, if it does potentially increase the risk of this condition, how would we monitor that? Well, obviously it's asking the patient if they have any changes in their urinary patterns, but I think sometimes it's always beneficial to maybe check a urinalysis and always follow that up with a microscopy, because just because you have hematuria doesn't mean it can't be something else due to like an infection, due to things like glomerular diseases, even though it's extremely rare, kidney stones. So always check that urinalysis with a micro to really take a look and make sure that it's truly like gross hematuria. And it's actually real, it's not pseudo hematuria. All right? So that's that part. Now, the next one here is we can kind of draw these. Let's actually kind of do this interestingly here. You see how the arms, they kind of look like a V? That's a reason for that. And this V is for the vinca alkaloids, all right? So you have things like vincristine, and you have what's called vinblastine, so and vinblastine. And these can actually damage the nerves, your peripheral nerves. And they can lead to something called peripheral neuropathy. Now, peripheral neuropathy, obviously when we talk about this one, you know, you're talking about potentially like loss of sensation, so decrease pain, temperature sensation, vibration sense, proprioception, and so they can even have pain like paresthesias as well. And so these are definitely things to be thinking about. And oftentimes it's just really important if the patient's on this to be asking them about that, but also be doing a very thorough neuro exam, doing your monofilament tests, getting out your tuning fork, banging that thing and checking to see if they have vibration sense, checking their finger position. Hey, where's my finger? Up, down, proprioceptive sensation, checking for fine discriminative touch. So making sure that you're doing a very good neuro exam is going to be important to identify any of these things. All right? And then, son of a gun, the next one that I want you guys to understand here is a little bit of a stretch, but it works. And this one is going to be kind of representing the bone. So what I want you to do is we're going to draw a 5, 6, a line, and then an MTX. 5, 6 lines, MTX. This is kind of trying to represent the femur in a way. And so this is going to be 5 is 5-FU. 6 is 6-mercaptopurine. And MTX is methotrexate. These act on the bone marrow. When they act on the bone marrow, so we'll represent it like this, it can lead to something called myelosuppression. myelosuppression. So myelosuppression is you can potentially drop your cell lines. What kind of cells? Well, the myeloid stem cell lines. So your white blood cells, your red blood cells, your platelets, they could all potentially drop, dude. And so what should you be watching for? I'd check a CBC to keep an eye on these patients, right? Now, there is other adverse effects that you guys definitely should be aware of. I mean, just because I'm mentioning these doesn't mean that there's other drugs that don't share some of these. I'll give you an example. So for example, we can also say that drugs like methotrexate can cause nephrotoxicity. That's a pretty common one. I could even say that things like your taxanes, which are also microtubule inhibitors, things like your docetaxel, paclitaxel, that can cause peripheral neuropathy. Oxaliplatin, which is a type of one of those platinum compounds of the alkylating agents, it can actually cause cold-induced neuropathy. So many of these drugs, honestly, almost all of these drugs any drug can cause myelosuppression. These just tend to cause the most severe myelosuppression. I mean, methotrexate can cause hepatitis. I mean, the list goes on. These are just the ones that you're most likely to get tested on, is what I want you guys to think about. Now, what I also want you guys to remember is when we talk about these drugs, there is this other category called immune checkpoint inhibitors. And this is something that it's really important. I used to see a lot in the ICU when you give immune checkpoint inhibitors, is patients would develop some issues where when you give these, yeah, you're giving it to target this types of reactions here. However, it's not uncommon for sometimes these monoclonal antibodies to attack other tissues. And if it attacks these particular tissues, they could develop unfortunate kind of like itises, right? The itis effect. So for example, if it attacks here, you could get something like colitis, you could get pneumonitis, you can get thyroiditis, you can get hepatitis. And so that's important to remember. So what could we see here? You could see things like colitis. What would that look like? I mean, it can vary. Obviously, it could be abdominal pain, maybe a low-grade fever, diarrhea. Here, you could have pneumonitis. This could be potentially, this could range. I mean, this could start off with just like shortness of breath, and then it could completely go full blown into things like cough, bronchospasm, hypoxia. This could be thyroiditis. And this could literally cause transient hyperthyroid symptoms. So they could have they could have a high thyroid hormone, right? And so that's another one to be able to think about here, they kind of present with like, with this one, it would have a transient effect. It's not gonna be long-term. And then the next thing is, well, if you actually do continue to cause these, if these MABs continue to damage the actual thyroid tissue, it can go from a transient hyperthyroidism to a permanent hypothyroidism. But either way, we'll talk about that on another day. And then damaging the liver, you could have hepatitis. And that's why it's actually very crucial when you put a patient on an immune checkpoint inhibitor to be doing things like checking their pulmonary function, checking a thyroid function panel, checking their LFTs. These are super, super crucial. All right. That really gives you the adverse effects. And when we talk about adverse effects, there's always some way, shape, or form that they try to kind of maybe weasel this into the exam sometimes. The real thing when it comes to detoxifying agents, I don't think for a lot of these that you'll get tested on them. I think that there's probably one, and that's going to be methotrexate. They may ask this question, and there's two reasons why, and I'll talk about it. Could you possibly be asked a question about other ones? Yeah, sometimes you could see things, maybe cyclophosphamide. I'd say it's less likely, but methotrexate, if a patient came in with things like severe hepatitis, myelosuppression, you should know what to give them. If they come in with hemorrhagic cystitis, you should know what to give them. Cisplatin, I do think that this is something to consider. Oftentimes it unfortunately leads to dialysis, but we should talk about this, especially if it's really bad enough nephrotoxicity. And then the last one is doxorubicin. So if you're kind of in that cardiology area, this may come up as well. But when we talk about these, the thing to think about here is whenever a patient gets methotrexate, and let's say that they develop some type of complication, myelosuppression is one to think about here. If they do, or hepatitis, we can potentially reverse that or kind of rescue the methotrexate effect with a drug called leucovorin. All right, so that's one to think about. Let me explain what I'm talking about here. All right, remember this pathway that we talked about 7 years ago at this point? It's coming back. So if you guys remember, we have something called folate, right? And then folate gets converted into dihydrofolate. That gets converted into tetrahydrofolate. That gets converted into 5,10-methylene. I'm just gonna put ME for right now, tetrahydrofolate. This was that pathway that we talked about. And folate, when it goes this way, there's a couple enzymes involved, right? This enzyme here is really important, and this is called, which one? Dihydrofolate reductase. That's the one that this guy inhibits. There's also this one called dimetylate synthase. What does he do? This is a good recap, right? He helps by taking things like your deoxyuridine monophosphate and converting it into deoxythymidine monophosphate, which we can incorporate into the DNA. And so whenever I use this, that helps to drive that process, yeah? Well, guess which drug inhibits this one? 5-FU. So 5-FU works here, and methotrexate works here. So what these do is these drugs actually work to inhibit these enzymes. So whenever we do that, what we know is, because we've already talked about this, we don't need to go crazy, it basically shuts down this pathway, shuts down the formation of DTMP and shuts down the DNA, right? If a patient's on methotrexate and we shut it down too much and we shut down that DNA synthesis enough that we're causing myelosuppression, we can give leucovorin. The concept behind leucovorin is that leucovorin is like a form of tetrahydrofolate. And so essentially what you're going to be doing is you're bypassing. So you're bypassing the enzyme. That's all you're doing. You're bypassing the enzyme. And so you're basically rescuing the patient. You're giving them this form of tetrahydrofolate. And then that's going to allow for them to go this way. It'll push them in this direction so that you can make more of this, so that you can stimulate this reaction, increase more of this, increase more. You see what I'm saying? That's kind of cool. And so that's the rescue process. So leucovorin will help to rescue methotrexate. However, if you give leucovorin, it actually potentiates the effect or enhances the effect of 5-FU. So that's what sometimes they'll try to test you on, is if you give leucovorin, what would it do to methotrexate and what would it do to the 5-FU? It'll rescue, right, the methotrexate and enhance the 5-FU. That's what I want you guys to remember. All right? All these other ones, cyclophosphamide, we can give particular agents. Doxorubicin, we can give particular agents. So if you really wanted to remember the cyclophosphamide, you can give something like Mesna, and I really focus on the fluids. Cisplatin, you can give things like amifostine, but I'd really focus on the fluids and then potential need for dialysis. And then doxorubicin, we can give something like dexrazoxane. Dexrazoxane. All right, so these are things that you can consider, but the really important one that I want you to remember is the leucovorin. All right, so this really, man, I mean, we talked about a lot in this lecture. We talked about the cell cycle regulation, the immune processes, the angiogenesis. We went through the cell cycle specific, the cell cycle non-specific antineoplastic agents. We went through the drugs that influence protein kinases. We went through drugs that influence the hormonal mechanisms. We went through drugs that influence the monoclonal antibodies. We went through drugs that influence the immune checkpoint. We talked about it in a holistic view. We talked about what are the combinations that are commonly utilized in different types of cancers. And then we finish it up with what are the primary adverse effects that you could be tested on, how to monitor them. And then the last thing is we finish off with what's a rescue med that we should know, especially in methotrexate toxicity. My friends, I really hope that you liked this lecture. I hope that you learned a lot. If you did, support us. Hit the like button, comment down in the comment section, subscribe. But my friends, I love you, thank you, and as always, until next time.